Splinter learning. Good morning to all everyone present. We are waiting for our judges to come. We'll start with the session soon. Good morning. A very good morning to everyone present here. I, Disha Shah, would like to welcome you all to the Day 3 of 3rd Student Research Congress organized by SVKM's Dr. Bhanuben Nanavadi College of Pharmacy, co-hosted by University of Mumbai, industry partners, ACG Worldwide and Cytec Center. The theme for 3rd SRC is Innovations in Healthcare. As quoted, innovation is unrelenting drive to break the status quo and develop a new where few have dared to go. For today, we'll be having two sessions. First would be an oral presentation competition by undergraduate students, followed by the presentation for Innovative Idea Challenge. For oral presentation competition under UG category, the judges are Dr. Clara Fernandez, ma'am. Ma'am is currently associated professor in Department of Pharmaceutics in Bombay College of Pharmacy. Ma'am pursued B.Pharm degree from SNDT University and a doctoral degree from ICT, University of Mumbai. She has more than 7 years of research experience and 20 publications, including research papers, review articles, and book chapters in journals and publication house of repute. She has one granted Indian patent with two filed patent to her credit and recipient of award research grants amounting to approx 1 crore INR. Ma'am has won second prize for poster presentation for collaborative research work titled Fabrication and Characterization of Camellia sinensis based wound dressing films for low suppurating wounds at one-day national seminar. She also won a Best Poster Award at SM Karandikar Award for research work entitled Impact of Drug Loading on Anti-Parkinsonism Effects of Lipid Formulation of Curcumin at South Asian Chapter of American College of Clinical Pharmacology. Ma'am has served as the Associate Editor of Control Release Society newsletter to a publication of CRS Indian Chapter. She is currently a member of editorial board of APTI Women's Newsletter. Our second judge for today is Dr. Nalini Kurup Ma'am. Ma'am is currently associated as an assistant professor in pharmaceutics at Principal K.M. Kunnani College of Pharmacy. She pursued bachelor's in pharmaceutical sciences from Bombay College of Pharmacy and master's in pharmaceutical sciences in quality assurance and PhD in pharmaceutics from CU Shah College of Pharmacy, SNDT University, Mumbai. She has more than 11 research publications. Ma'am has several grants received under her name. Grants received of approximate 1 lakh from University of Mumbai. Received grant of 17 lakhs from AICTE, RPS. She has filed an Indian patent entitled Novel Drug Delivery System comprising methotrexate. Ma'am has guided 11 M.Pharm students and is currently guiding 3 students. She is recognized PhD guide at University of Mumbai. She has served as a reviewer for Journal of Pharmaceutical Sciences and Technology Management and Journal of Cosmetic Dermatology and Indian Drug. She has a professional membership in Association of Pharmaceutical Teachers of India and Indian Pharmaceutical Association. Our third judge for today is Dr. Rashmi Malia, ma'am. Ma'am is currently associated as a professor and HOD in pharmacognosy in SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. Ma'am has teaching experience of 18 years and industry experience for 2 years. She has done her thrust research in areas of development of herbal formulation, phytochemical analysis, and biological evaluation of herbal drugs. She has received awards in consolidation for guiding a research proposal titled Moringa Tamarind Jellies, a new approach to combat helminthiasis infection in pure sciences category at postgraduate level at Avishkar Research Convocation 2020-2021. Ma'am has received Dr. P.D. Sethi Memorial National Award in 2016 for Best Research Paper on application of TLC, HPTLC in pharma, herbal, and other analysis. She has over 25 review and research papers published in many reputed journals. I would like to welcome all our judges with a boutique. A hearty welcome to all our judges.
Good morning to all the judges.
Good morning, ma'am, and thank you for the
Good
honor.
morning, ma'am. Yeah, thanks for the invite.
Thank you for accepting the invite. Thank you.
Most our pleasure, ma'am.
Thank you.
So before we begin with our session for today, all participants kindly take note of the instructions very carefully. Rename yourself as per allotted codes. Participants cannot reveal your name on any affiliation. Each presenting author will be given a time minute of 10 for presenting their presentation. Follow the time limit kindly and do not exceed. Only the presenting author will be allowed to answer in the Q&A session. Each participant is requested to stay alert for their roll call. As rightly said by Thomas Edison, the value of an idea lies in its application. With this, I would like to take permission from all our judges to start the session.
Yeah, we can start.
Yes,
And,
sure. And
yeah.
all the best to
And-
everyone.
Yeah, all the best. And I'm seeing that few of them have not renamed their this one joining IDs. Just see that, Nikita. Just make sure that everybody has renamed. Okay.
Yes, ma'am.
Prachi, Poonam, and all. So I don't know whether they are the participants or the guests. So just see. Huh?
Yes. Judges, I hope everyone has received their scorecard.
Yes, yes.
Yes, yes. Yeah.
Participant OUG1,
Yes, ma'am.
please be ready soon. We'll be presenting your slide.
Sure. Very good morning to all. So we starting our first project with research project with formulation and characterization of cubosomal in situ gel for fungal keratitis. So the first thing is content. What we are presenting? The first thing, introduction, rationale, aim and objectives, material and method, characterization, result and conclusions. So first thing,
Can you please
fungal
the slide? No?
Sure, ma'am.
Yeah, it's
Can you go to the first slide? We are
Sure.
on the 33rd slide is seen up to us.
Yes, hello. Can you start, ma'am?
no, I can't see the first slide.
The slide has not
Can you
changed.
share it? Yeah, can you please reshare the whole thing?
Sure.
Yeah, now it's proper.
It is visible, ma'am?
Yes, yes.
Yeah, yes.
Okay. Very good morning to all. The research project stated with a formulation and characterization of cubosomal in situ gel for fungal keratitis. The first thing we have introduced is
Put it on the slideshow mode.
slide on means presentation mode. Ma'am.
Slideshow, current slide करो.
Okay.
Participant OUG1, is it okay if we
No, it
present
is
your
my-
screen?
How it's okay, ma'am, if you present your screen from your
Okay,
side.
so we'll be presenting your screen soon. Kindly wait.
Sure. Sure, ma'am.
Your time starts now.
Okay. Very good morning to all. The research project stated with formulation and characterization of cubosomal in situ gel for fungal keratitis. Next slide, please. The content are introduction, rationale, aim and objective, material and methods, characterization, and result and discussions. Next slide. Next slide, please. Okay, the first thing, fungal keratitis. The fungal keratitis is obtained induced from a different species of bacterium and viral fungi. Normally, keratitis is inflammation of eyes cornea, which is called keratitis. So it is named as a fungal keratitis. It's a primary effect of corneal epithelium and stroma, and also in the anterior chamber of an endothelium of eye. This fungi can invade from stromal tissue to the effect of epithelial cells. The problem within this disease is it normally is upon Asia, specific Asia and East Africa and West Africa. Is high cases of fungal keratitis have been seen. Next slide, please. Next slide, please. Okay, so what is the cause of fungal keratitis? Means how patient having a problem during the keratitis. The main four factors are causes of fungal keratitis are underlying eye diseases, which is normally from a diabetes or something of injury have been recent eye traumas, weakened immune system, or frequently using of contact lenses. Is normally frequently using contact lenses, which is a normally a medicated, which has been inflammation or some problem regarding drug. Next slide, please. Okay, now the things are actually present. Means the medications and the therapies are present for the fungal keratitis are solutions. Means normal you can call that means eye drops. Means problem regarding solutions are rapid drainage from lacrimal tissue. Then second is suspension ointment. It cause blurring of vision and irritation of eye of some type. Normally the formulation of eye preparation doesn't have an irritation of eye, but sometimes the suspension cause irritation in eye. And third thing is lens and occlusal. First thing, lens is not or any of most of people are not aware of wearing that thing. An occlusal is need a specific doctor attention for insertions. Next slide, please. So, what is the meaning of why we want to make a cubosomal in situ gel? The first thing is considerably safe and stable, higher bioavailability, retention time of drug is high. The first thing for ocular formulation is that the drug has to be remain as such or at the place for a higher time for better effect. Third thing is thermodynamic stable for prolonged release, enhanced drug corneal permeability, and protection from encapsulated drug from physical and chemical degradation. The cubosomal gel also have a great load amount of drug owing and cubical structure with high stability and biocompatibility. Next, please. The aim and objective. The aim is for formulation and characterization of cubosomal gel in situ gel for fungal keratitis. The objective is to first thing a pre-formulation study, optimize cubosomal formulation by statistical design, characterization of evolution of cubosome, characterization evolution study of cubosomal in situ gel to perform in vivo study and accelerated stability study for prepared formulation. Next. These are the materials for preparing cubosomal gel. The drug is miconazole, which is also known as an antifungal gel. Lipids are glyceride monooleate, surfactant 280, and polymer is carbobol 934 for preparation of gel, and other are triethylamine. Next slide, please. So for formulation, there are two type of method for preparation of cubosomal gel. The first is top-down approach, and second is bottom-down approach. Top-down approach is normally goes with uh, general to specific approach, while bottom-down approach goes from the specific to general approach. Normally, in top-down approaches, in this method, we have taken a glycerine monooleate and our stabilizer with having stirring speed around 1000 to between 1500. Aqueous phase have been created. It is have to pass through high pressure homogenization to create a cubosome or nanoparticle, which also characterized by different studies. Next slide, please. This is a characterization, which is a preliminary study. Means, which type of concentration and which type of how much or different uh, ingredient have to be used. In this, we can see the lipid concentration is we use 3% different percentage. Surfactant, three different surfactant has been used. Surfactant concentration also has been varied, and homogenization speed is also varied for a batch to batch. Next slide, please. This is a box line can design. Means normally for staining design we have used there's a two type of variables. One is independent and second is dependent. In the independent variable, we are taking the concentration of lipid, concentration of surfactant, and stirring speed, which having a three major markings of low, -1, medium, and high one. And particle size is normally range from 100 to 300 nanometer. Enterprise efficiency is 50 to 80%, while drug release is 2 to 3 hours, which maximum we can achieve is a 4-hour drug release with for a in situ gel. Next slide, please. Next slide, please. So for after preparation of cubosomal gel, we have to check how it is prepared for which we are using a particle size, data potential, and point viscosity index. Then entrapment efficiency, surface morphology by SAM screening, and in vitro drug release study with in vitro drug release with a rotation of 100 rpm with using a type 2 dialysis bag method. Next slide, please. The preparation of cubosomal gel using has been used with a cooled method, in which we have used carbopol 934 with a 3 different concentration, chitosan with a 3 different concentration, and plutonic F127 with a different concentration. In this, the carbopol is a main ingredient for preparing a gel, which because it create a good stabilized gel, and also have a good viscosity of gel. Next slide, please. This is a pre-formulation study, which for miconazole is a calibration curve has been achieved from the UV. Next slide. This is an FTIR for miconazole, the drug we received from Mahasi Labs Private Limited, and the second image is describing a reference FTIR for miconazole. DSC of miconazole API and reference DSC graph for miconazole, which is describing the standard melting point is 100 to 102 degrees Celsius, while observed melting point is 85.24 to 85.29. This is an FTIR for miconazole, the twin 80, our suspectant, and the physical mixture, which is miconazole and a twin 80 mixture. The FTIR has been stored.
You have
Next
only
slide,
2
please.
minutes left.
Sure. Next slide, please. This is the effect of lipid concentration, is how the lipid concentration varies with particle size and PDI. We can see the different concentration have a different particle size, while we can see the 2% concentration giving a particle size of 117.4 with a standard deviation of 0.26, while surfactant we use is a different concentration having a different formulation, for example 188, for example 407, giving a particle size of different concentration, while we using twin 80 gives a 107 point standard deviation from 0.265 particle size nanometer with PDI of 0.749, standard deviation for 0.0455. Next slide, please. Next slide, please. Then the thing is, effect of homogenizer speed. Means, in this approach, the homogenizer is quite a valuable thing for having a good particle size, in which we can see the different speed has been used for a different particle size. We can see that the 15,000 RPM is giving a good speed, a good particle size of 143.1 plus standard deviation 0.87 with PDI of 0.296 plus or minus 0.02. Next slide, please. The selection of lipid for surfactant and concentration. This is just a preliminary study. Means which type of lipid have to be used or which combination have to be used. In this, we can just analyze which combination is giving a good particle size and good PDI for our formulations.
Your time is up. Now
Okay.
you're up for Q&A session with judges.
Sure.
UG1, you can just give the conclusion.
Sure, ma'am. For cubosomal gel, we have been achieved a 4-hour drug release with having a drug release about 93.24 to around 83 point something percentage, which achieve around 4 hours. So, ma'am, rationale behind this formulation is that normally the formulation we use, which is a regular therapy for eye or ocular release, means solution, ointment, and suspension, which actually discarded by lacrimal tissue, lacrimal drainage, or means it is also patient incompliance during that. Drug release have been very low for reaching to the surface of the eye ocular tissues.
Nalini ma'am, are you going first?
Hello.
Yes, ma'am.
Is
Yeah.
Nalini ma'am asking first or should I ask first?
You can ask, ma'am.
Okay. I had a query on, can you go to slide number 17? Okay, on slide number 17, you have mentioned that the melting point of your drug, okay,
Yes, ma'am.
was
Yes, ma'am.
not kind of, it's quite different from the standard. Okay,
Yes, ma'am.
and there's a
Quite different.
considerable difference. So, what was the reason why you have gone ahead with this drug then?
Yes, ma'am. The sample as a gift sample we received from a Maharshi Labs Private Limited. Miss actually, miss we use the DSC. They show us that can be pure impurity, ma'am.
So then how do you justify the assay or the concentration of drug you have used in the formulation? If you're saying it's impurity, you're getting a considerable difference in the melting point. So, how do you justify going ahead with this drug? It's failing in the melting point assay, melting point determination.
I'm not
Okay,
aware of
second question, I have a question for you is that, can you just give me a comment on the release mechanism of your drug from your formulation?
ma'am. The release mechanism of cubosomal gel is in situ gel is diffusion, ma'am. Diffusion.
So you got diffusion, because you got carbapol, you got pluronics,
934.
you got
Yes, ma'am.
monostearate, you got tween 80. So I was
Yes,
very
ma'am.
curious to know exactly what is the diffusion anticipated by you. Second thing for ocular formulation, what is the
Yes, ma'am.
estimated time for release? Like how long do you expect the release should happen? Because you showed later for 4 hours.
Yes, ma'am.
So how long do you
Ma'am,
anticipate the release should happen?
Ma'am, for we anticipated around 5 to 6 hours have to be drug release should be for a ocular drug release for better effect and for better therapy also.
What would be the residence time of your formulation?
Normally with the patient compliance, the formulation have to be residence around 2 to 3 hours easily, and it will not affect the visuals of a patient. Is normally the patient which having a fungal keratitis also have a problem with visuals, but the formulation we are using is quite transparent with having a gelling agent. Carbapol 934 give us a clear gel which not cause any visual impairment for the patient.
And why do you think a formulation will reside in the eye for such a long time? What is the mechanism?
Well, for in situ gel means normally is change. First thing is the formulation is having in a solution form. After having a temperature changes, it's around it is around body temperature is 28 degrees Celsius around, which formulation is apply as a solution. After that, it change from solution to gel formulation. That's the main reason of using in situ gel.
So what is the final component, gelling component? Pluronics and chitosan, or pluronics and carbopol, or just pluronics? What is the final
Means,
component?
final component is in carbonyl 934.
Okay. Thank you. Now I'm done. Thank you.
Nalini ma'am, you have
Yeah, sorry, I just got disconnected in between. I'm sorry if I'm repeating any question. How much time does it take for the formulation to form an in situ gel?
Around 43 or standard deviation with 1 second. 43 seconds. Around.
43 seconds.
Yes, ma'am.
Okay, so within that time, would any of the this would be lost, the liquid that you're applying to the eye?
Yes, ma'am. Normally, the formulation we thinking is that comes with a collapsible tube, ma'am, in which the patient have to be applied on the eyelid of the eye. After 43 seconds, the formulation converted into gel like formulation, which actually resides for a longer period, and it is actually means very convenient for patient to
So
use if we are
your
using eye
formulation,
drop.
your formulation is
Yes.
like a liquid viscosity or kind of semi-solid? Your final
Ma'am,
formulation.
final formulation, ma'am, is a kind of gelling, which is also called a semi-solid.
Okay. So it is already of a slightly higher viscosity, which increases in situ. That is what you're trying to say?
Miss, ma'am, the viscosity is not much that higher. That means if the viscosity is that much higher, the patient having a feeling of there is an eye which is patient incompliant. It is quite medium viscosity having of the formulation last
Okay.
formulation
No,
we have.
since you call it an in situ gel, that's why I asked.
Yes.
And another thing you had mentioned in advantages, it will have higher bioavailability. So what is the
Yes,
kind
ma'am.
of higher bioavailability that it will have? Have
Sure,
you
ma'am.
seen any research papers which report how much increase in bioavailability happens with your dosage form type?
Then the problem with the ocular drug is that the formulation does not resident on a ocular tissue. Man. Problem is that with using a cubosomal gel, we increase the residence time. If the residence time has been increased, the drug is have to be longer than normal usually therapies. Man. I mean, the problem
So do you get
with yes, ma'am.
okay. Do you get miconazole gel in the market? Plain gel?
Ma'am, is micronazole gel is means micronazole cream is available. The gel I didn't
What about
know
gel?
about much. I didn't know much about that gel, micro gel, which is used on a ocular tissue.
Okay, fine. Thank you.
one question from my side. Why cubosome specifically?
Well, the cubosomal gel, the using thing cubosomal gel having is high drug. The first thing is stability. For ocular tissue, the first thing is stability. Then is very low side effect, which I also regarding to the gel, ma'am. And third thing, ma'am, having high load capacity of drug to carry more drug than other formulation. And means that if we use another thing, means another formulation or another nanovascular system, that they might have causing something disruption in a visual impairment of patient. So that's why use of cubosomal gel, which is good stability and slightly no problem within using and applying or having good drug loading.
Any other novel formulation tried for eyes?
The recently means the normal therapies are using our means the normal therapies as
No,
we
no,
know
novel
that
drug delivery system. My question is any other novel drug delivery system tried for the eye with
means
the same drug?
the thing when thing is scaffold as used, but that much not quite known for that thing. Eye scaffolds, which is upon the frontal corneal epithelium layer, upon is have to be used upon corneal epithelium layer.
Okay, that's all.
Thank you.
Good work, actually.
Thank you.
Participant OUG2, please be ready. Will be presenting your presentation soon. Participant OGU2, you're not audible. Can you please check your mic? You're still not audible. We cannot hear you. Participant OUG2 will call you later. Participant OUG3, please be ready. Is participant OUG5 present? I repeat, is participant OUG5 present?
Yes, ma'am.
Will be presenting your presentation soon. Please be ready. OUG5.
Okay, ma'am.
Your time starts now.
Hello, ma'am. Hello.
Hello, yeah, you can start.
Good afternoon, all. I am presenting you the formulation and development of chlorthalidine loaded nanostructured lipid carrier. This was prepared by OUG5. These are the contents which are involved: rationals, material methods, in evaluation, result and discussion, conclusion, future scopes, and references. Introduction. Hypertension. As we all know, hypertension is a raised blood pressure in a condition in which blood vessels are persistently raised pressure. Blood is carried from the heart to the whole part of the body. Hypertension or elevated blood pressure is a serious medical condition to treat for the significant increases and the risk in the brain, kidney, and other diseases also. Chlorthalidone is a drug which is thiazide diuretic, which was used for the treatment of the high blood pressure. The bioavailability of the chlorthalidine is 65% due to the low solubility and low permeability of the pure drug. These are the conditions which are involved in the figure: hypertension. What are the problems with the drug and the solution? Problem with the drugs are the mainly their low permeability, poor aqueous solubility, lower the rate of dissolution, and lower viability. Solution is to improve the dissolution rate, reducing particle size, improve the dissolution rate by converting into the NLC, nanolipidic carriers, which are nano size, so that hello. Ma'am, presentation is not visible. Excuse me. Hello.
No, presentation is not visible.
Hello.
Yes, we'll be presenting it soon. Kindly wait.
Okay.
Yes, you can. Now it is visible. You can start.
Okay. These are the structures of SLN and NLC. SLN, which is solid lipid nanoparticles. NLC is the nanolipidic carriers. First generation of the solid lipid nanoparticles are like a brick wall model or brick structure. That's why incorporation of the drug is less as compared to the NLC. NLC is a structure like a stone wall model. That's why we can incorporate more drug into it, and the drug loading will be improved. Next slide. Rationale behind this study was viability of the chlorthalidone is 65% due to the low solubility and low permeability. By this NLC approach, drug dissolution rate was improved due to the lesser particle size as a nano level, and also improved the incorporating of the drug into it. Toxicity is less compared to the nanoparticles. NLC will give the better chemical and physical stability, and NLC will control the drug release, contain less amount of surfactants compared to the nanoemulsions. Materials and methods. Materials are involved in the procedures are solid lipid, liquid lipid, surfactant, and co-surfactant. Solid lipids are the glyceride, monostearate, cambritol, ATO, pacitol, AT05. Liquid lipids are oleic acid, castor oil, megalolol, A12, and surfactants are Twin80, polexamer, PVP, K30. Co-surfactants are used is glycerin, solutol, HS, Twin20. Method. For the preparation of the NLC, we are using ultrasonic evaporation method. Solid lipid and liquid lipids contained in the beaker, heat it into 60 or 70 degrees Celsius. Surfactant and co-surfactant then added. Mix the both phase and stir it at 2000 rpm, which is 2000 rpm, which is evaluated by box Bengen design for the 10 minutes. Then emulsions are prepared. then sonicate it, and LLC are obtained. Next slide. Evaluation. These are the evaluation parameters, which are FTIR, DSC, solubility study, particle size, and zeta potential, which are determined by zeta instrument. DSC, which is performed under the nitrogen flow. FTIR scanning, FTIR, which having the scanning range of 450 to 4000 centimeters square. Entrapment efficiency by separating the free drug sample from the solution. And putting the value on the equations. Then the in vitro drug resolution profile performed at the lab level normally, and same scanning electron microscopy, which shows the morphology results. Results are the pre-formulation study, identification of drug excipient compatibility study, different scanning colorimetry, Fourier transfer, infrared spectroscopy. Drug identification study, which involves the identification of the drug by UV, by the calibration curve. Calibration curve of the clothalidone drug was conducted under the 1.2 pH buffer and 276 nanometer wavelength, which is observed in UV. These are the values and calibration curves. DSC of the drug, DSC of the pure drug is shows in the first figure as the peak observed as a 2 to 8.59 degrees Celsius, which is corresponding to the to its melting point. Next slide. FTI study. FTI spectra for the reference and obtained drug are compared, and both are compared. Then the results show that the functional group matches with the standards, like stretching and nitrogen secondary group, imine group all. Next slide. Okay, these are the reference and FTIR study and the pure drug obtained pure drug FTIR study. Next. Okay. The drug excipient compatibility study. Drug excipient compatibility study of drug glyceride monostearate 2080 and change the slide, please. Castor oil, physical mixture, glycerin, and 2080. In this study, physical mixture and the pure drug shows the prominent peak. DSC, it shows the prominent peak of pure drug, which is 2 to 8.59 degrees Celsius, and the physical mixture shows the 179.58 degrees Celsius, which is corresponding to each other. And the temperature, when the temperature drops from the 2 to 8.59 to 179.58 degrees Celsius, the drugs start contact with the lipids, and peak shift will be observed. Next slide, please. Okay, these are the compatibility with GMS, castor oil, 2080, and glycerins. Next slide. Okay, these are the physical mixture study of the FTIR. Drug and physical mixture, there are almost all the functional group are the present, so there are no significance occurs. Next slide, please. Selection of solid lipid, liquid lipid, concentration of the solid lipid, liquid lipid, selection of the co-surfactant and co-surfactant concentration, and stirring speed, which will be analyzed. Next slide. These are the figures. On the basis of these, we can analyze like liquid lipid in the second slide, castor oil, oleic acid, and mygolol 8 0812, in which the particle size and entrapment efficiency considered to be considered as a variable, in which Casterwell shows the lower the particle size and good entrapment efficiency. That's why it will good for the formulation. Next slide. All are this likewise. Hello. Hello.
Yeah, we can hear.
Next slide. Next slide, ma'am. Okay. Optimization by the box bank and design. As the box bank and design involve low, medium, and high value, the amount of liquid lipid, amount of solid lipid, and storing speed is the independent variables, which are x1, x2, and x3. Dependent variables are the y1 and y2, which is particle size and entrapment efficiency. Constraints are showed in the particle size is 150 to 400 nanometer and more than 60% entrapment efficiency. Okay, these are the results of the box bank and design. Next slide. Okay, and these are the ANOVA applied on the particle size and ANOVA applied on the entrapment efficiency results. Next slide. Okay. Next slide, ma'am. In vitro drug release. In vitro drug release, in which the drug release was found. Drug release was found sustainably, and in chlorothalidine, the pure drug, the drug release will be nearly 56 to 58%, which was improved to 86%, 86.73%, which is better drug release. In vivo.
OUG5, your time is up. Now you're up for a Q&A session with judges.
Can I go ahead with the question?
Yes, ma'am. Sure.
Yeah, just one or two questions. First is, what is the BCS classification of your drug?
BCS class 4.
Class 4. Okay, and compared to NLC and SLN, why did you go in for NLC and not SLN?
Because SLN shows the bricks method, like drug incorporation is less as compared to NLC.
Okay, and one last question, why did you go in for box Benken design?
Hello, ma'am.
Why did you select box Benken design for optimization of your preparation?
For the ma'am, laser the run.
For the?
Hello. Less run,
Yeah.
and the variables are few. That's why. Which is the response surface.
Okay, fine. Thank you.
Okay.
I'm done.
Thank you, ma'am. I had just one question for you. Can you please explain how glycerine acts as a co-surfactant?
Hello.
Hello, can you hear me? How does glycerine act as co-surfactant? What is the mechanism?
No idea, ma'am. No idea. Sorry. I will surely take your point into account.
Thank you.
Okay.
Okay, this is nose to brain. You had done this, no?
Hello,
Nose
ma'am.
to brain delivery. For this formulation, how are you going to give this formulation?
Hello, ma'am.
Yeah. Can you hear?
Hello, ma'am.
Hello, yeah.
Hello.
OUG5, can you hear?
Yes, ma'am. Yes.
So, why did you choose? Your delivery was nose to brain, no?
Yes, ma'am.
So, how do you justify that delivery system for hypertension?
Sorry, ma'am. No idea.
Okay, okay. That's it from my side.
Okay.
Just OUG5, can you just keep the camera on for 1 minute? Or I can't see.
Ma'am, its video isn't supporting the browser. That's why it requires the download Teams.
Okay.
That's why the video isn't support.
Okay, yeah, fine. I think, yeah, that's it.
Okay.
You can call the next participant then.
Participant OUG2, please be ready. We'll be presenting your presentation soon. Is OUG2 present? I repeat, is OUG2 present?
BPS.
Next level. Is participant OUG3 present? Response. Last call for participant number OUG3. Are you present? Is participant number OUG6 present?
Yes, ma'am. Am I audible, ma'am?
Yes, you're audible.
Sure. Okay, ma'am. My present research is about pharmacological screening of medicinal plant Tinospora cordifolia and its silver nanoparticles against urinary tract infection causing bacteria. Urinary tract infection UTI, one of the most frequent bacterial infections after the respiratory infection in humans that affect any part of the urinary tract. Second most common bacterial infection. Two types of UTI, uncomplicated and complicated. Uncomplicated mostly occurs in female, it can be cured. Complicated, it occurs both female and male, it cannot be cured and leads to a therapeutic failure. Tinospora cordifolia, a medicinal plant rich source of antimicrobial activity and overcome the antibacterial resistance accepted by the antibiotics. These plants are used for treating the fever, urinary disorders, dyspepsia, syphilis, rheumatism, tuberculosis, rheumatism, and jaundice. The secondary metabolites are alkaloids, glycosides, deterpenoid, lactose, steroids, terpenoids, and aliphic components. Nanotechnology, a fastest growing area in the field of science, which is an interdisciplinary field of both science and technology. Silver nanoparticles were synthesized from aqueous silver nitrate through a simple and eco-friendly method using a leaf broth of Tinospora cordifolia as a reducing and capping agent. Silver nanoparticles are useful for the medicinal application due to their strong antibacterial activity. My aim of the research is to screen the antibacterial activity of Tinospora cordifolia and the silver nanoparticles against UTI causing bacteria. Objectives: to isolate and identify the urinary tract infection and causing bacteria from urine sample, to prepare the crude extract from the leaf by using the various solvents, to screen the antibacterial efficacy of the various extract prepared from Tinospora cardifolia against isolated UTI causing bacteria by using well-used diffusion method, to identify the phytochemical constitution present in the Tinospora cardifolia by GCMS analysis, to synthesize the silver nanoparticles from Tinospora cardifolia by characterization of UV visible spectroscopy, XRD, SEM, FTIR, and EDOX, to determine the antibacterial activity of silver nanoparticles against UTI causing bacteria. Methodology: collection of plant leaves authenticated washed in the tap water in order to remove the dirt and then dried under the shade dry condition for 10 days. Preparation of the solvent extraction by using the solvent extract method using petroleum ether, methanol, ethanol, ethyl acetate, acetone, and chloroform. Collection of urine sample. Urine sample was collected from government hospitals and isolation by using bacteria, a specific medium, UTI agar medium in a spread plate technique. Identification of UTI causing bacteria, microscopic examination, gram staining, negative staining, motility staining. Plating a nutrient agar in a blood agar, mechanical agar, and selective medium and biochemical testers. GCMS analysis of Tinospora cardifolia. By this method, BB5MS capillary standard and nonpolar column by using the volatile column. Helium gas was used as a carrier gas. Synthesization of nanoparticles by mixture of aqueous extract preparation by using the leaf or powder and 100 ml of distilled water, 100 degrees Celsius 20 minutes, and preparation of silver nitrate, 1 millimolar silver nitrate solution was prepared by distilled water and covered with aluminum foil. Synthesis of silver nanoparticles by mixture of these two, 10 ml of aqueous plant extract was added into a 50 ml of millimolar nitrate for the reduction into a silver ions and kept at room temperature for a week. This picture represents A, silver nitrate solution, and B, silver nitrate solution with Tinospora cardifolia extract. C, synthesized silver nanoparticles after a week. Synthesization of silver nanoparticle are characterized by UV visible spectral analysis, XRD, X-ray powder diffraction, IR, Fourier transform infrared spectroscopy, SEM, scanning electron microscopy, EDOX, energy diffusive X-ray analysis. Determination of
Yes, class. પછી દીકરી એમ કે કઈ ડીલ લેવી હે નોસ્ટ ટુ બ્રેન
Ma'am?
પછી નોસ્ટ ટુ બ્રેન
OUG5, can you please mute yourself? OUG6, please continue.
Yes, ma'am.
OUG6, we can't hear you.
Participant number OUG6, can you please unmute yourself?
Ma'am, is I'm audible? Identification
Yes,
of UTI. Okay, thank you. Identification of UTI causing bacteria. 5 bacterial isolators were identified: Staphylococcus aureus, Escherichia coli, Klebsiella pneumoniae, Proteus millabris, Pseudomonas aeruginosa. From this research, antibacterial activity of ethanol extract from by using the solvent extraction, Tinospora cardifolia against UTI causing bacteria, which is high in Proteus millabris in the zone of formation and less in Escherichia coli. Methanol extract from Tinospora cardifolia against using UTI causing bacteria, which is high in Pseudomonas aeruginosa and less in Staphylococcus aureus. By acetone extract of the leaf against the UTI causing bacteria, which is high in Staphylococcus aureus and less in Klebsiella pneumoniae. Antibacterial activity of chloroform extract from the Tinospora cardifolia against UTI causing bacteria, which is high in Proteus millabris and less in Escherichia coli. The ethyl acetate extract of leaf against the using bacteria, which is high in Escherichia coli and less in Proteus millabris. By this, by these research, best antibacterial activity of ranking is in methanol as the first ranking by using this, and acetone as the last one. Acetone antibacterial activity of methanol extract from the Tinospora cordifolia against using.
Continue, your voice is not hearing. Yes.
Ma'am, am I audible, ma'am?
Yeah, you are audible. OUG 6, continue.
Yes, ma'am. Yes, ma'am. This was a research which methanol as first place and acetone is very less in the antibacterial activity against UTI causing bacteria, which methanol extract from the Tinospora cordifolia against UTI causing bacteria, Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Klebsiella pneumonia, Proteus milliarvis in the zone of incubation. GCMS analysis of Tinospora cordifolia, which the peak represents 20 bioactive components of the Tinospora cordifolia. These are the 20 bioactive components of Tinospora cordifolia. UV visible spectroscopic analysis of synthesized silver nanoparticles. The maximum peak was found to be at 341 nanometer, as indicated that reduction of silver ions. Scanning electron microscopic SEM analysis synthesized by silver nanoparticles. According to the SEM image, the different particle sizes were observed. These was found to be at 15 nanometer and 20 nanometer and 50 nanometer. The energy dispersive of X-ray analyzes spectrum, EDOX, which is synthesized by silver nanoparticles. EDOX spectrum analyzes the reported that silver nanoparticles are using Tinospora cordifolia leaf extract was surrounded by a thin layer, some capping organic material form and extract. FTIR analysis of silver nanoparticles by using the peak identified that terrestrial alcohol methoxy hydrocarbon CF2 methyl carpetol. Antibacterial activity of synthesized silver nanoparticles against using UTI causing bacteria, which is high in Klebsiella pneumonia and less in Staphylococcus aureus. This plating represents this antibacterial activity of synthesized silver nanoparticles against UTI causing bacteria, Staphylococcus aureus, Escherichia coli, Pseudomonas aeruginosa, Klebsiella pneumonia, and Proteus milliarvis. At conclusion of my research, the outcome of present research concluded that the pharmacological study carried out an Indian medicinal plant having traditional claims of effectiveness might warrant fruitful results. Tinospora cordifolia serves as a useful source of new antimicrobial activity and phytochemical which can act biochemical components. When compared to the other solvents, the methanol extract of Tinospora cordifolia has showed maximum antibacterial activity against UTI causing bacteria. Nanotechnology is the vigorous research area in the current science field. Silver nanoparticles have considered that application in the field of medicine, mechanical, chemical, electronic, optical, food, agriculture, and environmental. Comparatively, silver nanoparticles have shown maximum antibacterial activity than methanol extract of Tinospora cordifolia. Thank you, ma'am.
Thank you, participant OUG 6. Now, the session is open for Q&A.
Clara ma'am, yeah, Nalini ma'am, shall I ask first?
Yes,
Yeah,
ma'am.
sure.
Go ahead.
So you have done GCMS of your Tinospora cordifolia, right? OUG 6?
Yes, ma'am.
Yeah.
Yes,
so
ma'am.
why GCMS specifically?
Ma'am, GCMS is specifically for identification of bioactive components present in the leaf, ma'am.
So what are there any reports which say, so what is GCMS analysis helping you? How is it better than your HPTLC? You could have done a HPTLC or a HPTLC, no? Why GCMS specifically?
Ma'am, gas chromatography mass spectrometry, ma'am. It is for gas, helium gas is carried as a carrier gas, ma'am. HPLC we have not done.
Yeah, okay. So from GC, what constituents might be responsible? Did you come to know from the GCMS? Which constituent of Tinospora might have good antimicrobial property?
Ma'am, we are more efficient more efficient, ma'am, than other tests, ma'am.
Okay, one question, one more question. This extract, which extract was the best, you said?
Methanol extract is best reducer, ma'am.
And when you did
Because
your, when you,
antimicrobial activity against UTI causing bacteria, ma'am.
correct. So methanol extract was good, no? So when you prepared
Yes,
your silver nanoparticles, why
Yes,
did
ma'am.
you use which extract?
Ma'am, to get a specific what are the components present in this, by this we can treat other medicines, ma'am.
No, which
No
which
powder
ex
can no
you
powder
use
can be used.
powder or you use methanol extract?
No powder, ma'am. We used methanol extract, ma'am. From
Methanol.
powder, we used methanol extract for silver nitrate, ma'am.
You used powder and then or methanol extract. What did you use?
Ma'am, first we have used powder, then we extracted methanol, ma'am.
Methanolic extract you prepared and that you use for the formulation of silver nanoparticles?
Yes, ma'am. Yes, ma'am.
Did you check the silver? Silver itself has a good antimicrobial property, right?
Yes, ma'am.
So did you do a comparison of?
Yes, ma'am. The silver nitrate has antimicrobial activity or efficient.
So did you do a comparison between silver nanoparticles, simple and plain, and the other one, loaded one? Did you do a comparison of that?
No, ma'am. We have not done, ma'am.
Okay, okay. That is it from my side.
Can I continue?
Yes, yes.
Yeah. Like ma'am mentioned, you have not compared with plain silver nanoparticles, blank silver nanoparticles. Similarly, you should have compared. Have you compared with methanol also? Only methanol as a solvent?
No, ma'am. We have never compared at methanol as a solvent, ma'am. In the mixture of the powder we used, ma'am.
Yeah. So
The
when you
extraction.
made the when you did your diffusion assay in the Petri plate, one
ma'am.
well could have been for your plain solvent to show that your extract has more activity.
Yes, ma'am.
Right?
Yes, ma'am.
Yeah, and another thing, when you have taken the UV for silver nanoparticles, have you compared
Yes,
it with
ma'am.
the UV of blank, only the extract? Have you done UV of the extract before converting it into silver nanoparticles?
No, ma'am, we have not done, ma'am. After the silver nanoparticle extraction, then we have done by UV visible spectroscopy, ma'am.
Yeah, because you're saying that peak which was got is of silver nanoparticles. So how do you ensure that it is of silver nanoparticles only? UV just shows you a peak, right?
Yes, ma'am. By article reference, ma'am.
So this work has been reported?
No, ma'am. We are under still in process, ma'am.
No, you just said you referred. So if it is not recorded, then how did you refer for the UV lambda max?
Silver nanoparticle article, ma'am.
Okay, fine. Thank you.
Thank you, ma'am.
I'll extend the question asked by Rashmi ma'am and Nalini ma'am with regards to your MIC studies. In your plates, you have not anywhere taken the standard drugs. So these bacteria which you have isolated, were they resistant ones or they were vulnerable to antibiotics?
It is sensitive, ma'am. It's sensitive to the antibiotic, ma'am. Sensitive to antibiotics, ma'am.
Do you have a report saying that all these bacteria which you have isolated, they were sensitive?
Yes, ma'am. Yes, ma'am.
Okay. Because the reason is that most of the UTI bacteria have resistant problem. That's the reason I'm asking you this particular question. And second thing, on slide 13, you're showing slide 13, you're showing a color change after a few days, I believe.
Yes, ma'am.
So, yeah. So your students, yeah. So how do you justify this? After 1 week you've taken, right?
First, we have taken silver nitrate solution, ma'am. Then silver nitrate in the mixture of cardifolia extract, ma'am. Then after a week, it was synthesized as silver nanoparticles.
So what is the entire protocol as to how it becomes nanoparticles? It takes 1 week for making a nanoparticles, or it's instantaneous? How is it?
Ma'am, after a week, we are testing this. We concluded that silver nanoparticles was present in this leaf.
So did you do an optimization, time the process?
No, ma'am.
And did you analyze it for particle size or something?
Yes, ma'am. I have analyzed it, ma'am.
Okay, so now when ma'am had asked you a question about the UV, remember you
Yes,
mentioned about
ma'am.
UV. So can you somehow give a correlation how particle size and the UV will be able to, correlate?
Ma'am,
You
I
read,
have.
you read articles on nanoparticles, right? That's what you mentioned.
Yes, ma'am. Ma'am, UV analyze.
How does UV data correlate with the particle size of silver nanoparticles?
Ma'am, UV. I have analyzed, ma'am. The maximum peak was found to be at 341 nanometer and indicated that reduction
No,
of
no,
silver ions in my slide
no, you got that. You got
29,
that.
ma'am.
You got that. What I'm asking you something. When you have particle size, silver nanoparticle, UV is one of the way to correlate the nanoparticles dimension, the change, So, because you read the articles you mentioned, right? You've read articles and literature on silver nanoparticles. So, is there
Yes,
any
ma'am.
mention where UV can be one of the tool to assess the change in particle size of silver nanoparticles?
Yes, SEM, ma'am. Scanning electron microscope.
No, no. I mean, there is a change. When you have a change in particle size, the entire UV changes. Coming to your again to the antimicrobial studies, just show
Yes, ma'am.
me one of your plates. In most of your plates, I saw that the zone of inhibition is not distinct. Yeah, you can take this
Yes,
only
ma'am.
one, anyone, Pseudomonas. It's not distinct.
Yes, ma'am.
Do you think you can consider the data? Even for Staphylococcus aureus, your
Yes, ma'am.
zone of inhibition is not distinct. It is merging. Do you think this is an accurate data?
No,
Can we conclude on this data?
ma'am, by using different amount, it can be differed, ma'am. For 50 mg, it can be differed,
No,
75 plus
I'm
100.
asking you a
So you
question.
place.
See, it's not that. I'm
Yes,
asking a
ma'am.
question that your zone of inhibition is merging, right?
Yes, ma'am.
So when it's
Yes,
merging,
ma'am.
can we consider this data? Aren't
Yes,
we
ma'am. We
supposed
can
to replace
conclude it, ma'am. We can conclude, ma'am. Everything is same. The amount of given which injected in the well was different, ma'am. That's it, ma'am. We need that sensitive or resistance to the bacteria.
Okay. Then, thank you.
Thank
Ma'am,
you,
I'm
ma'am.
done. Thank you. Yeah, thank you.
Thank you, participant OUG6 and judges.
Hello.
Participant number
Sorry for the
OUG7.
hello. I'm OUG2. Can you hear me?
Yes, we can hear you.
Can I present right now or I will wait for the end presentation?
Yes, sure, you can present your slide.
Okay. Thank you so much. Sorry for the delay. I was facing some problem regarding audio.
Will you be presenting the slide or should we?
It will be good if you present it.
Okay, we'll be presenting your slide shortly.
Okay, thank you. Okay. Good morning, everyone. The
Your
topic
time
of
starts
today's Okay. The topic of my presentation is formulation and characterization of ellagic acid encapsulated surface modified polymeric nanoparticles for oral cancer. Next slide, please. These are the contents: introduction, anatomy, statistics, causes of oral cancer, conventional treatment and its problem, aim, objective, material methods, evaluation, result and discussion, conclusion, and references. Next. Now, we all know that cancer is a very deadliest disease, and recently it is the number of cancer cases were increasing day by day. So it requires attention of healthcare providers to form, as a pharmacist also to provide some formulation which can have the effect or positive, a good effect on cancer treatments. And among the cancers, oral cancers are very prominent in our India also, and in worldwide also. Uh, in oral cavity, the area or the part in which oral cancer occurs are the tongue, then tissue lining the mouth and gum, under the tongue, at the base of the tongue, and the area of the throat at the back of the mouth. Next slide. Next slide, please. Okay, so in the first image, you can see that the first half is normal cells, and the second part is starting of the cancer, and the third is the full-fledged oral squamous cell carcinoma. So this is the anatomy, and in the second figure, you can see the oral cavity, and inside that we have discussed the tongue, on the tongue, then lining of the mouth, then base of the tongue. So these are the part at which oral cancer can be occur. Next. These are the statistics. First statistic is regarding the site, like which site is more likely to produce oral cancer. So you can see that 20% having floor of the mouth. So on the floor of the mouth, localization of cancer will occur, on the 21% anterior base of the mouth is there. And on the second statistic, you can see that oral cancer has 56.4% of chances among all the cancers, and it has a 50-50 ratio of survival rate. Next. Now, causes of oral cancer. These are the more prominent causes like tobacco and metal chewing, smoking, alcohol consumption, and HPV infection, and other causes. And other causes are age-related, then your diet and lifestyle also. Now, for the treatment, we have surgery, chemotherapy, and radiation. But as a pharmacist, we have to consider the chemotherapy treatment, because in the surgery treatment, reoccurrence chances are happen. In surgery, they are removing the cancer cells with the normal cell lines to ensure that no any other cells were remaining. But still, it has a chances of reoccurrence, and it causes eating problems as it is related to the oral cavity. And the radiation therapy has also its own disadvantages. In chemotherapy, we also have some disadvantages. So we will discuss in next slide. Next. Okay, so we have several chemotherapeutic drugs, citizumab, cisplatin, paclitaxel, methotrexate, etc. Like we have around 20 anti-cancer drugs, and we are using from many years. Like in TB treatment, also we have some of drugs, and we are using from like around 30 or 40 years. As the topic suggests, that innovation in healthcare. So the cancer is also the newer formulations which have less side effects and more bioavailability. So we have two routes: oral administration and intravenous administration. Oral route is preferred by the patients, but compared to IV route, it is less effective. So if we go with the IV route, it will have benefits, but still both the routes have different disadvantages. So the solution is nanotechnology. Next. Okay, so the main objective regarding this study is to apply the nanotechnology, and we are adding natural drug plus biodegradable polymer. And the third thing is we are acquiring the targeting approach. So for the nanotechnology approach, we are formulating polymeric nanoparticles in natural drugs. Like in these recent years, people are focusing on the natural remedies or natural drugs because it have less side effects, but still they have a problem. Like my drug is allergic acid, it is a polyphenolic compound, and it is stated it is in class 4, so it have a problem, but still it has a anti-cancer activities. So we for to overcome that barrier, we are introducing nanoparticles and polymers. So in polymers, bovine serum albumin we have taken because it is readily biodegradable, and it is not harmful for our body or and also the environment. And the targeting approach, so we are giving the predefined address to the drug that you have to go to that particular site only, not anywhere else. So it will not cause any cytotoxicity to the other tissues or other organs. So for targeting approach, we are targeting the folate receptors, and for that we are using folic acid. Okay, aim and objective are covered, but the other thing is we are also introducing the QBD based approach in our formulation. Next. Okay, material methods. So ellagic acid, bovine serum is polymer, butyraldehyde as a cross-linking agent, ethanol as desolvating agent, and folic acid as a targeting agent. Next slide. The method which we are using is desolvating agent, in which we are taking drug and polymer protein in one beaker, and we are adding ethanol simultaneously to form the aggregates, and then we are cross-linking it with butyraldehyde. Next. After that, this complex, we are adding folic acid, and the folic acid drug and polymer complex will happen. And at the end, folic acid conjugated bovine serum albumin nanoparticles were formulated. Next. Evaluation parameters, UV visible spectroscopy, DSC studies, FTIR solubility studies, then PDI, entrapment efficiency, zeta potential, XRD studies, SAM, in vitro and evoviability studies, and stability studies. Next. Okay, so this is the calibration curve, and the lambda max was 253 nanometers for the ellagic acid. Next. Okay, this is a drug and excipient compatibility study. So in first graph, the very first peak from the left side, it's indicating the 3559, the peak 3559 peak, and in the second, the peak was 3557, and that peak is for phenolic compound, and that is the main identification point for our ellagic acid drug. And rest of the you can see that rest of the peaks were also similar with the drug only. So we can say that there is no any such kind of interaction is occurring between drug and excipients. Next. Okay, now solubility studies. It is important because we are using desolvation technique. So in which we have two solvents, like one is good solvent, second is bad solvent. Good solvent means it will solubilize our drug, and second one is it will precipitate out our drugs, and that is on basis of dielectric constant of particular solvents. So when we evaluated, ethanol has higher like it has a low dielectric constant, so it indicates that when a small amount of ethanol is added into the solution, then then protein interaction will be increased, and aggregation will occur, means precipitation will occur. So that's why we have chosen ethanol as a desolvating agent. DSC studies, in DSC studies, the peak was the melting previous one.
We have 2 minutes left.
Okay, next. Next slide. In QBD-based approach, we have QTBP elements which are dosed nanoparticles, then targeted drug delivery, IV route, and higher Cmax and AUC. For that, we have the critical parameters are particle size, entrapment efficiency, zeta potential, and PDI. Next. Yes, for assessment, we have formulated Ishikawa fishbone diagram. So from material, methods, and machines, you can see all the 8 parameters which are important for the formulation. So further, we are going to screen out by Plackett-Bowman design. Next. In Plackett-Bowman design, we have chosen these are the 8 factors which were shown in the Ishikawa diagram, and for the screening purpose, we have added in the Plackett-Bowman design. Next. For that, we have generated Pareto chart in which among that 8, we found 3 are very significant, like amount of bovine serum albumin, amount of resolvating agent, and pH of bovine serum albumin. So that for the further optimization, these three factors were put in the box Venkan design. Next. Okay, the contour plot and response surface. So contour plot and response surface for particle size were formulated. In contour plot, amount of pH of the solution and amount of bovine serum albumin were taken, and in that we can see as as the amount of bovine serum increases, the particle size will decreases, and as the pH increases, the amount of drug will be entrapped will be more, and also the particle size will be less. Same was response
Your
surface
time is
plot
up.
was. Okay.
Your time is up, and now you're up for Q&A session with judges. Thank you.
Okay.
Yeah, hello.
Ma'am, should I go?
Yeah, Clara.
Thank you. Can you please comment on the stability profile of ellagic acid?
Uh, uh, Sorry, ma'am, the stability was not performed due to some reason.
No, but what is the ellagic acid? It is it pH dependent solubility? Does ellagic acid have pH
Yes,
dependent
yes,
solubility?
yes, yes. Yes, it is soluble. It is soluble in a higher pH. Like we required buffers, like
What is the pH of the buffer?
around 7.4.
Okay. So this, what formulation you're designing, you're intending it to give it by intravenous route, am I right?
Yes, yes.
Okay. So my question to you is that when you talk about bovine serum albumin and we're talking pH 7.4, how will it ensure that drug is protected in the blood?
Drug is protected in the blood.
In the blood. How will albumin nanoparticles ensure that drug is protected in the drug? It has low bioavailability, na, this molecule?
Yes, yes, yes. It's class 4. Yes.
So how would it ensure that drug is protected in the blood? And how will you ensure that the drug is reaching in maximum amount to the oral? Because if you recall in
Like,
biopharmaceutics, so
yes.
no. Oh, you go ahead, go ahead.
So we have performed in vivo bioavailability studies. So in that we found that T half is 8 hours, and like us all this data from that we are saying that it is protected, because otherwise if not protected then we see the burst release or some kind of effect, and in vitro release studies also.
So do you think in vitro release gives you a clarity about how it's going to behave in vivo?
Uh, uh, in vivo, ma'am, it is actually a joint work, so the quality part I'm not much aware about it.
No, just I'm asking because when you do parental, you need to also do the other interaction, no? Because you've got albumin. Okay. Second thing, since this is just a proof of concept, okay. When I give intravenously, how do I ensure that more drug reaches the oral mucosa? Why did you think of using a buccal patch or something like that? Why did you choose to go for an intravenous formulation?
Because we are adding the targeting agent, so it will ensure that the drug will reach the target. That's why.
But it's oral cancer, right? So oral cancer, will it not be
Yes.
accessible to us? Do I need to go via intravenous route to deliver drug which can be easily accessible? This site?
For the patches, we have to choose the buccal route, buccal patches and all. So, but
So wouldn't that have been
comparatively,
a better option?
sorry.
Wouldn't that have, I mean, when you go intravenous route, the first organ which will get the blood supply in large quantity, your drug will go there. So oral does
Yes.
not become the first priority. So definitely, and you say folate receptor.
Yes, but
So my
oral
question
has
is
the
that,
problem with.
yeah, go ahead.
Oral has the problem with the toxicity and all those things, but comparatively, oral IV will be more preferable.
Okay, I'm done. Thank you. Nalini ma'am, you can go and answer ahead.
Okay, one question from me. This ellagic acid, why did you choose ellagic acid? And did you do
Because,
any cell line studies?
no, ma'am, cell line studies were not performed. But in the literature reviews, we have find that ellagic acid is emerging for the anti-cancer activities, and it is used widely. Research is going on for the breast cancers, prostate cancers, and colon cancers. So we decided to choose the oral cancer for the implementation of ellagic acid nanoparticles. So from the literature itself, we found that it has anti-cancer cancer properties, and it has good amount of mechanism. It increase the tumor suppressor genes like P53 and P21, and so that's why we choose that.
So these, the P53 and also these are prominent in oral cancers, or
yes.
means how can you say so? Is there literature which states that ellagic acid is good in oral cancer? How is it?
Yes, yes. But targeting approach was not added in that, so we decided for in literatures we didn't found, and if and the folic, especially for folic acid.
Okay. The folic acid was a target receptor. Means what is it?
Yeah,
Why did you add
folate receptor.
folate receptor? It's there in the
Yes.
oral cancer.
Yes.
Okay. Yeah, that's it.
Yeah, just one question from my side. Since you're talking of a parenteral preparation, what can you comment about the sterilization aspect of the preparation?
Means the end product is our particles, nanoparticles, not the suspension one. Nanoparticles were there, but in animal studies we have given in vivo, like in IV route.
Yeah, IV route. So do you need to sterilize it or no?
Like a reconstitute powder. Like first we have it will
Yeah,
separately tag
agree.
like in a particle.
Yeah. So whether separately or later reconstituted, will an aspect of sterilization come into picture?
Yes, means the suspension in the solution which we are using, that it should be sterile. And then we are
Yeah.
adding our
Yeah.
particles.
How will you sterilize it?
Sorry, ma'am. I don't
Okay,
have
fine.
an
Thank
idea.
you. Yeah, thank you. Done from my side.
Thank you, participant number OUG6. Now I would like to call participant number
I,
OUG7.
excuse me, I guess she was OUG 2, right? Can you
Yes, she
just
was
Yes,
confirm?
OUG2.
Yes,
and OUG2.
yes.
Finished
She
before
was
her.
OUG2, sorry. Participant number OUG7, are you there?
Yes, I'm here.
Do you want us to share the presentation or will you
Yeah,
share?
okay. Yeah, okay.
So we'll be sharing presentation from our side. You can start the presentation. Your time starts now.
Can I start?
Yes, you can.
Yeah. Good morning, one dot. My topic is antifungal potential of medicinal plant extract Cassia arata against fungal pathogens. Cassia alata, ornamental medicinal plant of Cassia Phenoidae family, traditionally used for the treatment of fungal disease, particularly skin-based superficial infection, commonly called as ringworm plant. It's very famous for treating ringworm and other skin diseases. It also have antibacterial activity, antiviral property, anti-inflammatory, antioxidant, antipyretic, anti-helminthic, anti-diabetic, and anti-tumor properties. It's highly effective against the opportunistic pathogens such as Candida albicans and dermatophytes, like Microsporum canines and Trichophyton mentagrophytes. The present study was aimed to study the antifungal potential of medicinal plant extract against fungal pathogens. And my aim of this study is antifungal potential of Cassia alata against fungal pathogens. My objectives: prepare the extracts from Cassia alata leaves by using solvents, collect the fungal pathogens, Candida albicans, Microsporum canines, and Trichophyton mentagrophytes, and screen the antifungal potential of Cassia alata by agar-well diffusion method, and analyzes the bioactive compounds present in Cassia alata by GCMS, and formulate the coconut oil-based Cassia alata lotion for treating fungal infections. And my methodologies. First, I collected the Cassia alata leaves and shade dried the collected leaves, and I did succinate extraction using ethanol and acetone. And collect the fungal pathogens, Candida albicans, Microsporum canines, and Trichophyton mentagrophytes from MTCC, and determination of antifungal activity of Cassia alata by agar-well diffusion method, and analysis of bioactive compounds in ethanol extract, Cassia alata by GCMS analysis and FTIR analysis. And finally, I formulated the coconut oil-based Cassia alata lotion for treating fungal skin infection. And my results from this tabular column, Microsporum canines shows Microsporum canines is highly sensitive to this Cassia alata plant, followed that Trichophyton mentagrophytes, and Candida albicans is less resistant. From this, ethanol shows high inhibitory activity and acetone is less inhibitory activity. And GCMS analysis, we have 13 bioactive compounds. 1 butanol 3 methyl, 1,6 anhydro beta D glycopyranase, 3O methyl D glucose, oxyrane, 10 methyl E11 diene, 1 ol propionate, 1 aspartic acid, 2,6 dihexadenoate, 14 methyl 8 dexylene 1 ol, oleic acid, vitamin E acetate, 1,2 bi-trimethylsilyl benzene. From this FTIR analysis, we have 21 bioactive compounds such as alkyl aldehydes, aromatic sulfones, sulfonyl fluorides, sulfates, sulfonamides, alkanes, aromatic alkenes, ester, allenes, chitines, isocyanates, isothiocyanates, acetylene, nitrile, paspine, aldehyde, alkane, amide, alkanes, carboxylic acid, and alcohol. And this picture is the Cassia alata plant. And these are the ingredients I used in formulation: Cassia alata leaf powder and coconut oil. And this picture is the mixing of ingredients. And this is the boiling of ingredients. And this is the final product as skin lotion. And my conclusion: Cassia alata is widely used as traditional medicine in India. In many rural areas of our state, Cassia alata is used to treat various skin diseases caused by bacteria and fungal infection, and also for insect bite. In conclusion, Cassia alata in combination with coconut oil will be the good remedy for fungal skin infections. And also, I personally experienced that in past year, I was infected with ringworm infection. On that time, my grandma suggested this plant, and also I get positive result. And this is the good remedy. And thank you.
Thank you, participant number OUG7. Now you're up for a Q&A round with judges.
Yeah, OUG7, just 1 or 2 questions. Did you check for the antifungal activity? Have you checked for the antifungal activity in the lab?
Yes, ma'am. Yes, ma'am.
How did you do that?
By well diffusion method, ma'am. Okay. Sorry, ma'am,
Okay.
I can't hear you.
Against which organism did you check? Which organism did you use to check the antifungal activity?
Candida albicans. Microsporum, ma'am.
Only one? 1 or 2?
Sir, ma'am, I did 3, ma'am.
Okay. And how did you prepare this extract, Cassia alata leaf powder?
Ma'am, salt rate extraction, ma'am, using ethanol and acetone.
Ethanol and acetone.
Acetone, yes, ma'am.
Okay. And whatever components you got in your analysis studies that you did, are they reported in literature? Did
No,
you
ma'am.
compare?
No, ma'am.
Okay, fine. Thank you.
Thank you, ma'am.
Yeah, shall I ask the question?
Yes, ma'am.
This cassia alata, you did 2 extracts, right?
Yes, ma'am.
And when you prepared the lotion, what did you do? How did you prepare the lotion?
Ma'am, I used leaf powder and coconut oil, ma'am. Then I boiled.
Why did you use the leaf powder? And you had done the extracts and checked for the antifungal activity with the extracts. And you
Ma'am, it's
showed one
highly
of them is
effective.
good. Yeah, and then why did you use the leaf powder then? Why did you use the leaf powder?
Extract is, ma'am, it have high bioactive compounds, ma'am.
Yes, but when you are preparing the lotion, you could have used the leaf extract na, instead of the powder. So, and show me the lotion photo. Show me the next slide. Yeah. So, when you use the powder and coconut oil, do you think you will get such a lotion? Or did
Yes,
you
ma'am.
use an
Actually, I
extract?
did it, ma'am.
Hmm?
Actually, I did it in lab, ma'am.
Yeah, okay. So, what is the preparation method of lotion of with coconut oil? What did you do?
Ma'am, first, for these are the ingredients, ma'am.
And then what did
Then
you
I
do?
mixed.
It is simple mixing.
Yes, ma'am. It's simple mixing, and also I have personal experience, ma'am. I use this
Okay.
lotion for my infection, ma'am.
Okay, coconut. Why did you use coconut oil?
Ma'am, coconut oil also have some beneficial, ma'am.
What
We
benefits?
can also use water. Coconut oil is a carrier, ma'am, for this lotion.
Okay. That's it from my side. Clara, ma'am.
Yeah. What is the definition of lotion?
Sorry,
How do
ma'am,
you
I
define
can't
a
hear
lotion?
you.
How do you define lotion as a dosage form?
Ma'am, in gel form?
No, you're mentioning this as lotion, right? But you just have your powder and the oil. So, on what basis are you concluding that the formulation can be termed as lotion? So, how do you define lotion?
I mean, finally it turns into gel form, ma'am. Then I consider this as a lotion.
And why does it form a gel? You don't have a gelling agent in your formulation, right? So, why does it form a gel?
In future we will extend this work, ma'am.
No, okay, fine. Anyway, thank you.
Thank you, ma'am.
Yeah, you can call the next participant.
Thank.
Disha, you can call out the next participant. Who is it?
Participant number OUG8. Are you present?
Yes, ma'am, I am present.
Will you be sharing the presentation or do you want us to share?
Yeah, I will share, ma'am.
Okay.
Ma'am, is the screen is visible, ma'am? Hello.
No, it's not. visible yet.
You share already, but it's not. Um, you share it, ma'am.
Okay, we are sharing it.
Hello.
Yes, we'll be sharing it soon. Your time starts now.
Ma'am, still I didn't get the screen, ma'am.
Is the screen available? Can you see it?
No, ma'am. No, ma'am. It's not visible, ma'am. No, ma'am. I didn't see it.
I think OUG herself can then share the screen.
Ma'am, I have shared before itself, ma'am, but let me try again, ma'am. Is this visible now, ma'am? Hello, ma'am. Is this visible, ma'am?
Yeah, we can see.
Visible, ma'am.
Yes.
Is the slide already? Hello?
Are you sharing the screen OUG8?
Yes, ma'am. I am sharing, ma'am. I have
I
shared already, ma'am, but
request you
the
to
screen is not
please
opening.
stop sharing the screen because it is not visible from our side. We'll be sharing the screen.
Oh, you are going to share, ma'am. Ma'am.
You're sharing. I kindly request you to please wait.
Yeah, okay, ma'am. I will wait.
Judges, sorry for the delay. We are facing some network issues from our side. I request you to please be patient. OUG8, have you stopped sharing your screen?
Yes, ma'am. I have shared already, ma'am.
Okay.
Ma'am, still my slide is not visible, ma'am. Ma'am?
Can you please wait for a
Ma'am?
moment?
Yeah, okay, ma'am. Yeah, okay. I will be waiting, ma'am. Okay.
Okay, OUG 8, can you share your
Yes, ma'am.
screen?
Ma'am, I have shared already, ma'am, but the someone. Okay, ma'am. I will try again.
Yeah.
OUG 8, I request you to share the screen from your side once again.
Ma'am, is it visible, ma'am?
Share and share it.
Ma'am?
OUG 8, unshare whatever you have shared before, and then again you share it. Try doing.
Still no. Ma'am?
Participant number OUG 8 will call you later. Your slide is not visible from our end. Participant number OUG 9, I request you to be ready. I repeat, OUG 9, I request you to be ready. OUG 9, do you want us to share the slide or will you be sharing?
Yes, ma'am. Please share it. Good morning. Am I audible?
Yes, you're audible.
Yes.
Your time starts now.
Yeah. Good morning, everyone. So the title of my presentation is Quality Standardization and Phytosome Formulation of Trigonella phoenum glycum. Please, can you change the slide? Hello. Yeah, the contents are introduction, the goal of the project, and we have done the phytochemical, microscopic, and pharmacognostic evaluation. Then the extraction process of the seed or the plant we have collected. Then phytosome preparation, evaluation of the phytosomes, conclusion and reference. Please, can you share this slide? Change the slide. Yeah, introduction. We have made a novel approach for manufacturing of the pharmaceutical goods that overcome the limitation of the conventional dosage form. The hydrophilic compounds, which are mainly less marginal effect and poorly absorbed because they have larger size and low lipid solubility. So we have made this approach to increase or overcome these side effects of the conventional dosage forms. Please, can you change the slide? Yeah, the phytosomes are the novel dosage forms which are mainly designed to improve the bioavailability, therapeutic efficacy. These are formed by reaction between the phytophosphatidinil and the plant extract in the presence of the aprotic solvent. These natural formulations of the drug delivery system is in which the lipid surrounds or the phosphatidyl chloride of the lipid binds to the herbal extract of the phytoconstituent to increase the absorption or the bioavailability of the herbal drugs by the pharmacodynamic and pharmacokinetic parameters. Please, can you change the slide? Please, can you change the slide? Yeah, the drug of our interest or the Main active ingredient which we have extracted is from the Trigonella phoenum graecum, which is commonly known as fenugreek or methi. We have selected this drug as this drug is used from the traditional, from many years it has been used because of its various pharmaceutical action. It has got other nutrients aspects also, such as calcium, zinc, and iron, and many fibers. The extract of fenugreek from the literatures, we have seen that it has got the chemotherapeutic actions, and it is also used in the manufacture of the steroidal products. It has got the hypoglycemic effect and hypocholesteremic effect, and due to phenolic and flavonoid properties, it has also antioxidant property. The goal of the uh, project was to standardize the Trigonella phoenum graecum seeds for quality analysis and to develop the novel dosage phytosome to overcome the side effects of the conventional dosage form. Please, can you change the slide? Yeah, we have first done the microscopic evaluation to check or to identify or make sure that the drug is identified. The shape of the drug was rhomboidal or smooth shape. Size is 5 to 8 mm long and 1 to 2 mm thick. The color was light to dark yellowish brown. Odor was characteristic. Taste is mucilaginous and slightly bitter. Can you please change the slide? Yeah, the next study done was pharmacognostic studies where the all the basic pharmacognostic evaluations were done, like LOD, moisture content, ash value, and extractive value. It was compared with the standards that were found in IP, and the loss on drying was 8%. Total ash was also in limit. The extract, we found that the aqueous extract was major, like we can extract the drug in aqueous form, and even alcoholic has given good results. Can you please change the slide? Can you please change the slide? Yeah. Then the next phytochemical screening was done to identify the active constituents like alkaloidal test, glycosides, steroids, triterpenoids, flavonoids, and saponins. So various tests were done. Please, can you change the slide? Next, we went for the extraction of the seed. Extraction was done in hydroalcoholic extraction, where 95% of ethanol was used, and even it was followed as cold maceration, then the soft slate extraction. You can see here our extract. Yeah. Can you please change the slide? That was subjected for phytosome preparation, where thin layer hydration method was used. Soy lecithin and cholesterol was basic component to form that phosphatenyl chloride layer. 4 different ratios were selected. Firstly, the soy lecithin and cholesterol was mixed in 10 ml of chloroform, and the drug was mixed in ethanol. And then rotary evaporator was set, followed by the hydration, and then probe sonicated to reduce the size of the phytosome. Can you please change the slide? Can you please change the slide? Am I audible? Yeah. So there were 4 different ratios of cholesterol and soy lecithin that we selected to get the reduced size of phytosome for increasing the bioavailability. As we see, the F3 formulation has shown the less particle size, that is 180 nanometer, and we can say like the lesser size, the more bioavailability can be seen, as it can penetrate the biological membranes very faster. Yeah, can you please change the slide? The conclusion was we have standardized the herbal drug as it is important technique for assessment of the quality of the herbal drugs, and assessment of the quality. The Trigonella phoenum seeds was accomplished as we have done the studies on the morphology, phytochemical, and physiochemical evaluation, and the results were within the standards, which we have compared it with the IP. And then the present work aims to prepare the phytosomes to overcome the obstacles of the conventional dosage form, as it is novel lipid based nano formulation. These are the references. Thank you for giving me this opportunity to present. Thank you, ma'am.
Thank you,
Sonia, may I-
participant number OUG9. Now you're up for Q&A round with judges.
Ma'am, may I ask the question first?
Yeah, sure.
Thank you, ma'am. Can you just explain why is your encapsulation efficiency decreasing when the phospholipid concentration is increasing? The previous slide, not the previous slide.
Because the particle size also decreases, so the efficacy also decreases. Like in the phytosome, the entrapment is also less.
So with a higher concentration, you're having very low phospholipid. F1 has low phospholipid.
Yes, ma'am.
And still its PDA is quite high. We're getting entrapment is very high. And as you go on increasing the concentration of both cholesterol and soy lecithin, we are seeing a decline in the encapsulation efficiency. So what were you, according to you, which one is the best formulation to go ahead with? The low particle size or the high entrapment efficiency?
We would go for the particle size because we are mainly focused on the particle size as it has to cross the biological membranes. If for
what is
to
the ideal particle size looking at?
Sorry, ma'am.
What is the ideal particle size you're looking at?
We have not set like standard, like we are looking for this only. We have just we were looking for the lower particle size.
Okay.
Which one is lower? That one we have considered as best one. Can go for it.
Thank you, ma'am. I'm done.
Thank you, ma'am.
Yeah, one question from my side. What is the activity that you're looking at of Trigonella phoenum?
It has got various focused on the antioxidant activity, we can say, because if we take it orally, it is best suited for antioxidant activity.
Yeah, but antioxidant targeted for what?
Targeted for antimicrobial, but it will be further study. We have not yet conducted that study. Antioxidant for the antimicrobial.
Yeah, because you have developed a lipid-based drug delivery system, right?
Yes, ma'am.
And you were saying the penetration will be better with a smaller particle size.
Yes, ma'am.
So penetration where will depend on what activity you are giving it.
Yeah, ma'am.
So your preparation would be what? An oral route? What route would you give it?
Still, we have not decided for the route, but we are planning for going for the antimicrobial activity. But we have to conduct the test for that. I can't conclude it till then.
Okay, fine. Thank you.
This you said that this Trigonella methi seeds, na? So these are the limit, the phytochemical or the morphology and all are within the limits. So which limits or which standard limits have you followed?
We have followed the IP, ma'am. In the IP, what standards they have given, that we have compared with that.
In IP, you have methi.
Oh, yes, ma'am.
Okay.
And few of the literatures also we have gone through where they have given the standards and like just referred.
Yeah. One thing, if you want to do an antimicrobial activity, so do you have literature which states that methi is antimicrobial?
Yes, ma'am. Yes, there are literatures which states it has got antimicrobial property as well.
Which extract has good antimicrobial property according to literature?
Don't know.
Okay.
But methi has antimicrobial activity. We have seen that.
Okay, fine. But why phytosomes then if you want to do an antimicrobial activity with methi? Why phytosomes specifically?
Because phytosomes are they have good penetration power in the biological membranes, so we have chosen only phytosome. Lipid based, only if we take lipid formulations, there are the chances like the drug is released or not, we can't predict that. So phytosomes are now studied for, these are better than lipid formulations, they are said.
Which are the which lipid formulations are you talking about?
Ma'am, in previous slide, can you please change the slide? I have shown one diagram there.
You can explain. I don't. If you can remember.
Lipid, like totally lipid. If we don't take phosphatidyl chloride as a layer, only lipid layer. If we take, it has got some limitations. So we have chosen phytosomes.
Okay, fine, fine. Yeah.
Yeah. Thank you, ma'am.
That's it from my side also.
Thank you, participant number OUG9 and judges. Now I
Thank you, ma'am.
request participant number OUG8 to be ready. OUG8.
Yeah, ma'am, I'm ready.
So, do you want us to share the slide?
Yes, ma'am.
Okay. Your time starts now.
Yeah, okay, ma'am. Good Hello, everyone. Good afternoon to all. Good afternoon to all. The content of the presentation of my topic is in vitro antimicrobial evaluation of some substituted novel benzimidazole analogous. It defends that antibacterial and antifungal activities are studied by in vitro. Ma'am, slide is not visible, ma'am. Ma'am, slide is, yeah. This is the content of my topic. Introduction, literature review, aim and objectives, experiment part. In result and discussion, we are going to see chemical and biological part. At last, conclusion reference. Next slide. Let's come to introduction part. Heterocyclic system of benzimidazole is plays very important role in past and recent is because it's containing nitrogen containing molecule. Benzimidazole have biological activities like anti-cancer, anti-inflammatory, antimicrobial, anti-cancer, and anti-tubular. According to recent result, antibacterial mechanism is, ma'am, slide is not visible. The antibacterial mechanism of benzimidazole is due to similar compound. Benzimidazole replaces purine by blocking key component. Benzimidazole activity is possible by act through a new mechanism and it those effective in antibacterial drug in current use. Recently, much attention has been paid for development of benzimidazole containing antibacterial agents, which is lead to new development of molecule. Next slide. Yeah, let's come to literature view. In 2012, Gaidachari and team workers were reported antimicrobial activity. In 2013, Bharat and KP workers were reported antimicrobial activity. Next slide. Next slide. Yeah, in 2009 and 2011, Wasserman was reported antimicrobial activity on different substituted compounds. In 2013, YB was reported fungi activity in vitro method. Next. Ma'am, next slide, ma'am. Yeah, in 2013, Sharma was reported anti-tumor activity. In 2012, Vaccine was reported anti-cancer activity. Ma'am, next, ma'am. Next slide, please. Yeah. In 2012, Demira reported anti-cancer activity and anti-HIV drug and benzimidazole derivatives. And in 1985, Janssen's were reported antihistaminic activity of benzimidazole. This is the structures of the related compounds. Ma'am, next slide, ma'am. Ma'am. Yeah, let's come to aim and objectives. Benzimidazole is very much useful in molecular development and development of pharmacological screening. To synthesize a benzimidazole, it has varied biological and pharmacological activity. This derivatives possess anti-tubular, anti-cancer, antiviral, anti-inflammatory activities. In present study, we synthesize different benzimidazole group on first position by refluxation and stirring method. Ma'am, slide please. Ma'am, slide is not visible, ma'am. Ma'am. Yeah, the aim and objectives. These are the summarization of my project work. We are synthesizing a title compound, and we are going to add 5 derivative compounds. All compounds are purified by chromatography technique. New compounds are synthesized by characterized by analytical and spectral data. Analytical and spectral data include thin layer chromatography, infrared nuclear ionization, and mass. The synthesized compound are evaluated by antibacterial and antifungal activity to identify the active compound for further molecular development. Next slide, ma'am. Yeah, these are the mechanism of my scheme. Let me explain it. When phenylene diamine is reacted with formic acid at 100 degrees Celsius for 2 hours, we are adding sodium hydroxide to change acid to alkaline medium, and we are getting substituted benzimidazole. Compound 3 is substituted benzimidazole. Compound 3 is reacted with chloroacetyl chloride in presence of catalyst potassium carbonate. Ma'am, back, ma'am. Ma'am, I didn't complete the. Ma'am, I didn't still complete. Can you come previous slide? Yeah. Benzimidazole was reacted at 80 degrees Celsius as potassium carbonate, and we are getting fifth compound. Compound 5 is benzimidazole chloroethanol, and benzimidazole chloroethanol is reacted with potassium carbonate as a catalyst, and 80 degrees Celsius was reading with we are getting title compound benzimidazole ethanol. Next slide, please. Next slide. Next slide. Yeah, these are the physical data of my synthesized derivative of title compound and 5 derivative compounds. In physical data, we are added molecular formula, molecular weight, RF value, and infrared values, and melting point percentage. These are the physical data of my yield. Ma'am, next point. Next slide. Next slide, please. Ma'am, next slide. Hello, ma'am. Slide is not visible to me, ma'am. Yeah. We come to resultant discussion. We are going to see about two headings. One is but and other one is first we are going to see part. Higher spectrum of compound 3 indicate peak at 3,200 to 3,300 centimeter inverse. The compound 5 was intense peak for chloride is 757 centimeter inverse. For carbonate compound it was 1,660 centimeter inverse. The NH that compound 3 is disappear in 3,200 to 3,400. That compound obtained on different derivatives to the nucleophilic of compound 5 by adding and using different. It's the title compound disappearance of chlorine at 757. the NMR spectra of protein value and the protein peak up in the total value is 723 to 3.65. Next slide. Next slide. Ma'am. Ma'am, slide please. Ma'am, slide is not visible to me, ma'am. Ma'am. Ma'am, slide is not visible to me, ma'am.
Yes, we have changed the slide from our side.
Ma'am, it's not visible to sell, ma'am. Ma'am, these are the spectral data. Yeah, ma'am. These are the spectral data. These are the spectral data of nuclear magnetism and mass. And mass. These are the spectral data of the compound tree shows very clear and most intense figures observed in compound tree indicates most intense figures observed when the allergen compound protein translation. The allergen figures observed in one pack of intense, so it named as a dark peak. The growth was next slide. Please. This is the field of the compound by using NMR analysis. Please. The slide is not visible to me, ma'am. Ma'am, can you hear me, ma'am? Ma'am, can you hear me, ma'am?
Yes, we have changed the slide from our side.
Ma'am, but the is visible only, ma'am. Let's move to antibacterial. In biological, but we are using which dilution method? We are using gram antibacterial. Bacteria we are using focus. We are taking pseudomonas, adenovirus, and salmonella typhi unequal. The test of the culture for unincubated and grown 37 degrees Celsius. We are using dimethyl formamide as a short, but dimethyl formamide does not have micro growth of Mulla Hilton agar culture is using. Mulla Hilton agar culture is we are using for 37 degrees Celsius for 1 day. 1 day. We are using antibacterial activity. We are using amoxicillin standard for antifungal activity. We are using glycofulvin as a standard drug. In bacterial activity. Yeah, okay, ma'am. In bacterial activity, the PS3 compound is more potent, and compound 4 shows moderate potent, and compound 2 shows less moderate, and PS1 and PS5 shows very least activity because it's containing in fungal activity. PS3 shows very strong active, and PS1 and PS4 compound shows moderate active, and PS2 and PS5 shows very less activity. In 50 microgram. In 50 microgram, the concentration, the microgram should not show any growth for antibacterial and antifungal activity. When it is in 100 microgram, concentration very growth. And this is the representative and the graphical diagram and statistical diagram for me. And compound 5 shows very less activity, and compound 3 is very high potent, ma'am. Next slide. Yeah, we are coming to synthesis. The synthesis of BNCP substituted benzimidazole ethanol was prepared by stirring and reflux method. The title compound have activity antimicrobial activity. Among title compound, chlorophenylamino benzimidazole ethanol was the most active compound, which is more as equal potent to the standard amoxicillin drug. And interestingly, griseofulvin compound shows a equal and moderate compound, moderate to griseofulvin, and therefore we are and therefore serve as a lead molecule for further modification to obtain clinically useful novel classification of antimicrobial agent. Ma'am, next slide, ma'am. Ma'am, next slide. These are the reference of my thesis paper. Yeah, that's it, ma'am. Thanks for your kind attention, please, ma'am.
Thank you, participant number OUG8. Now you're up
Yeah,
for
welcome, ma'am.
Q&A round with judges.
Yeah, okay, ma'am.
Can I start?
Yes, yes.
Yeah.
Yeah, OUG8, you said you have used
Yes, ma'am.
griseofulvin as the standard drug, right, for antifungal activity?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Yeah, so can you comment on your compounds, whether analogs which they were comparable to that or better than griseofulvin?
It's equal, ma'am. It's equal. It's equal, near to equal, ma'am. The graphical representation diagram shows, ma'am. They are equal, ma'am.
Yeah, so have you done any statistical study or just from the graph you are?
No, ma'am. Yes, ma'am. Statistical diagram I have done.
So you tested
Yes,
it at a
Yes, ma'am.
particular
I have tested.
significance level? Which
Yes, ma'am.
which
I have
statistical
tested.
tool did you apply? Which statistical tool you applied?
Ma'am, antifungal and
What I
bacterial
mean
statistical.
is, yeah, but which
Ma'am?
statistical test did you apply which said that it is comparable to the standard drug?
Ma'am, 1 second. Ma'am, can you say the question again, ma'am?
Yeah, there are some statistical tests, right? Test for significance to find out whether your compound is comparable to the standard, better, or not as
Yes,
good
ma'am.
as the standard. So, which statistical test did you apply to study this? Okay, fine. Yeah, thank you.
Ma'am, just
So what is the toxicity profile of your synthesized compound? Have you done any
Ma'am, I
study on
didn't.
that?
Ma'am, I didn't.
What is the toxicity profile?
Ma'am, I have to go for further modification, further development. Just I have done only in vitro. I didn't do toxicity, ma'am.
So what is the log P of your molecules?
Ma'am?
Log P? Log P. How hydrophobic are your compounds? Are they comparable to griseofulvin or more?
Yes, ma'am.
Or less?
Yes, ma'am. Equal, ma'am. It is not less than, ma'am. It is equal to the drug, ma'am.
Okay. That's it. Thank you.
Okay. What
Okay,
about
thank you, ma'am.
the
Thank you, ma'am.
purity of your compounds? Did you check the purity?
I have analyzed the special data of 40 billion mass. Very clear.
Yeah, but is your compound free of all the intermediates or the starting material and all? So did you check the purity? What is the yield of your compound?
I checked all the drugs purity, ma'am.
How did you check the purity?
For the derivative compound 1, ma'am, by NMR mass. By NMR and mass technique, ma'am. And compound 1 yields 82%, ma'am. And compound 2 yields 86%, ma'am. And compound 3 is very, very active and it is yield 95% yield, ma'am. And compound 4 is 89%, ma'am. And compound 5 is yield very 60%, ma'am. Because compound 5 was aliphatic and aromatic, so it is give less activity, ma'am.
So you in a synthesized compound you check the purity with mass and IR and all?
Yes, ma'am.
Or
Yes,
you
ma'am. I
use
check,
any
ma'am.
chromatography to check the purity?
Yes, ma'am. Chromatography I didn't use, ma'am. Mainly I have used only nuclear magnetic resonance and mass like this, ma'am.
Okay.
Because in our college we are providing with some facilities, ma'am. So I have done only some facilities, ma'am.
Okay, fine.
Okay, thank you, ma'am.
That's it.
Okay, thank you, ma'am. Thank you, ma'am.
Thank you, participant number OUG8 and judges.
Thank you. Thank you.
Now, I would like to call participant number OUG10. OUG10, are you ready?
Yes, ma'am.
Are you going to share the slide or do you want us to share?
No, ma'am.
Your time starts now.
Good afternoon to all. So my title name is Formulation of Novel Phytosome loaded with the Trigonella Phoenum graecum extract and its brine shrimp lethality assay. So, ma'am, can you change the slide, please? Okay, so introduction. In today's world, the herbal medicines are becoming increasingly popular for their ability to treat the variety of diseases with less harmful effects and the better therapeutic values. So as we know that the herbal medicines are popular because of their less side effects. And the phytosomes are the novel dosage form that has been introduced for improving their bioavailability and therapeutic effect of the drug. So we selected the Trigonella phoenum graecum, commonly known as the fenugreek, which is belong to the family Fabaceae. It is the most promising medicinal plant that has been ever existed because it has the many medicinal values and the many biological properties such as it is antioxidant, gastroprotective, antimicrobial, anti-diabetic, anti-rheumatism, etc. And we mainly focuses on the antimicrobial activity of the trigonelline. It provides the natural food fibers and the other nutrients required in the human body. So mainly the goal is to formulate the phytosome loaded with the Trigonella phoenum graecum extract. First we perform the extraction, that is maceration followed by soxhlate, and that extract is then that extract is further studied for the formulation of phytosomes, and the prepared phytosomes are evaluated for the brine shrimp lethality assay, that is to evaluate the phytosome loaded with the Trigonella phoenum graecum for the brine shrimp lethality assay. So phytosomes are the novel drug delivery system containing hydrophilic bioactive phytoconstituents of herbs surround and bound by the phospholipids. The phytosome technology produces a little microsphere or the little cell which protects the plant extract or its active constituent from the destruction by the gastric secretion and gut bacteria due to the gastroprotective property of the phosphatidylcholine. Ma'am, can you please change the slide? Okay, so preparation of phytosomes. We have prepared the phytosomes by rotational evaporator technology, or it is also called as the thin film formation method. So the phytosomes were prepared by using the thin layer hydration method with the different ratios of drug, soy lecithin and cholesterol. The soy lecithin and cholesterol that were previously weighed were dissolved in 10 ml chloroform, and the drug was dissolved in the ethanol, and they are ultrasonicated for 10 minutes using the bath sonicator. Ma'am, can you change the slide? We use the rotary evaporator to form the phytosomes, and the temperature is set to the 45 to 50 degrees Celsius at the 180 RPM is used to form a thin coating of phospholipid, and the combination is developed. We have totally formulated the 4 formulations of with the different ratios of soy lecithin and cholesterol. Then the film was kept in a freezer for the 24 hours, and that process is called as the hydration, and that process is carried out to remove the excess solvent. That is, this film was kept in a freezer for 24 hours, then add 1:1 ratio of chloroform and ethanol, and then attach to the rotary evaporator for the hydration process at 180 RPM for an hour. Ma'am, please change the slide. Okay. Then following hydration, the lipid and the plant extract combination was prop sonicated for 30 minutes in an ice bath to dissipate the heat. After that, the Trigonella phoenum graecum nanophytosomes were manufactured, and they are kept in a freezer in amber colored bottle to protect from the light. Then 4 formulations with the different ratios of soalecithin and cholesterol were prepared, and the prepared formulations are evaluated for its following activity, that is particle size determination and entrapment efficacy. So we have concluded that the F3 formulation is showing the less particle size, that is 141 nanometer with the PDI 0.7, and the entrapment efficacy is 81%. So F3 is exhibited the good properties, hence the F3 is subjected for the further analysis for the biological properties. Ma'am, can you change the slide? Okay. So for the antimicrobial activity, we have done means we have to we have going to do the 2 activities, that is brine shrimp lethality assay and agar well plate method. But our research is still going, so only I have included the brine shrimp lethality assay. So brine shrimp lethality assay is a simple and inexpensive bioassay used for testing the efficacy of the phytochemical present in the plant extract. The present study determined that the extent of the lethality was directly proportional to the concentration of extract. It is based on the killing ability of the test compound or simple zoological organism, that is brine shrimp, and they are derived from the Artemisia salina. Ma'am, can you change the slide? Okay, so what is the procedure for the brine shrimp lethality assay? So the extract were routinely evaluated in a test for lethality to brine shrimp lethality larvae. Toxicities of the compound were tested at 1 ppm, 10 ppm, 100 and 1000 ppm in a test tube which contains the 10 ml seawater. Then the 10 brine shrimps were used in each chips, and the survivors are counted after the 24 hours. Then the blank control is used with distilled water, and the lethal concentration for the 50% mortality after 24 hours was determined using the probit method. This assay is widely used in the evaluation of the toxicity of heavy metal, pesticides, medicines, especially the natural extracts and extra. This method is very useful for the assessment of the toxicity of plant extract, and it is also used for the preliminary anti-cancer activity. So the conclusion of my project is, it can be concluded that the novel lipid-based nanoformulation loaded with the Trigonella phoenum graecum show the better result to the BSL bioassay. It may work as an effective model as an anti-cancer moiety. Further in vivo and clinical trials are required to support this work. The present may serve as a base for the investigation of the anti-cancer drug with enhanced therapeutic activity by means of a new drug delivery system. And okay, ma'am, thank you so much for giving me opportunity to present.
Thank you, participant number OUG 10. Now you're up for Q&A round with our judges.
Hello. Yeah. So
Hello,
what was the So what was the IC50 of your this one formulation?
Ma'am, we are not still calculated because ma'am, our project is ongoing, and so we have not calculated still.
Okay, but it has
only we have done the activity of BS.
You have done the activity? Continue.
Yes, ma'am. Only calculation is remaining. Ma'am, I can tell the means how many shrimps are survived in the test tube. Ma'am, we have taken 4 test tubes. In T1, the concentration is 10 ppm. In that, 7 brine shrimps are alive. And then in T2, the concentration is 100 ppm. In that, 4 brine shrimps are alive. And T3, we have taken the 1000 ppm. In that, all the shrimps are died. And T4 is taken as a standard. In that, 9 brine shrimps are alive. In T4, we have used the distilled water.
So there is no need of calculating the IC50 and all for your, because anti-cancer activity of methi, is it reported? Do you have reports of the anti-cancer activity of
Yes,
trigonella?
ma'am. Literature. Yes, ma'am. Literature
So
have
what was
that.
the IC50 reported there?
Ma'am, that I don't know, ma'am. Means I don't refer that. Means only reported I have see that.
That's it from my side. Nalini madam
can
or
you- Yeah, okay. Ma'am, go ahead.
Oh, that's done. From my side it is done. So you can ask.
How can you compare brainstorm assay with anti-cancer activity? I mean, any literature states that what the way it performs over there, you can conclude it might have anti-cancer activity.
Yes, ma'am. The articles are present on that brain shrimp lethality assay to prevent to present the anti-cancer activity. We refer from that only.
So, in what context do we? Because you've not compared plain phospholipid, right? You've not compared your blank
What, ma'am?
formulation along. You haven't compared blank formulation out here, right? Or plain extract. Am I right? In your brainstorm assay, it's just the formulation.
Use the phytosome formulations. Ma'am, we have
So
done
you
the
haven't
phytosome
compared.
formulation.
So you
What,
haven't
ma'am?
compared that with the blank. You haven't compared it with the blank, right? Plain
Yes, ma'am.
phospholipid and plain extract. You haven't compared, no?
No, ma'am. Only a 4 phytosomes with different ratio. That 4 phytosomes we have done compared with the BSL 40. Plain extract were not used.
So because you're making a comment that it can be explored further for anti-cancer activity, do you think we should also compare that with the plain extract and see?
What, ma'am?
Do
Can you
you
pardon,
think
please?
the study should? Do you think the study should include also plain extract? Since you're making a comment that phytosomes can really help, so before you make a conclusion, should we also try and check what happens with the plain extract or plain phospholipid?
No, ma'am. But in future,
Okay.
ma'am, still I
Thank you.
have not done with the plain extract. But in future, I will try, ma'am, with plain extract.
Yeah, thank you. That's it. Thank you.
Thank you, ma'am. Thank you so much.
Thank you, participant number OUG10 and judges. Now, I would like to call participant number OUG3. Participant number OUG3, are you ready? This is the last call for participant number OUG3. Otherwise, you will be debarred from the competition. Now, I would like to call participant number OUG4. Participant number OUG4, are you ready? This is the last call for participant number OUG4 or you will be debarred from the competition. I would like for the judges to make sure that participant number OUG3 and OUG4 has been cancelled from the competition.
Okay.
I would ask our judges to provide a brief message regarding the session to all our participants.
Hello,
Just
Clara.
to make an observation based on the presentations, first and foremost, all the works were exceptionally done, well thought of. For an undergraduate student, it was a very exemplary effort from each one of you. And as informed by, as suggested by all the judges, we expect you to, kind of enrich your data with few more studies and hopefully try and communicate and publish your work so that many more people are aware of your, the findings of your research, and that can help many more researchers. So, with this, I congratulate all of you because at this young age to make a presentation, research presentation, is commendable. So, all the best to each one of you, and wishing you great luck and for your future, a bright future. Thank you.
Thank you, ma'am.
Yeah, even I have the same thing to say at this, BPharm undergraduate level or undergraduate level. It is really good that you're doing such good work, and few things, yeah, as seen, and even the presentation skill, the knowledge on what research you have conducted, all that was good. Okay, so as Madam said, it is best that you publish this research work, whatever you have done. Yeah, so get that published. So you keep an aim of publishing these things. And so it is because this work which you have done is really good. Okay. So till that you publish and then only you can go from your undergraduate, you pass out. Okay. So keep that aim of publishing. That is it. Otherwise, all the knowledge or the presentation, everything was good of all the students. Yeah, that is it. Solution.
Thank you, ma'am. I would like the judges to receive a small token of appreciation from us. Thank you, Dr. Clara Fernandez, ma'am.
Thank you.
Thank you, Dr. Nalini Kurok, ma'am. And thank you, Dr. Rashmi Malia, ma'am.
Thank you.
Thank you to all the judges for providing us with such insightful suggestion and feedback to all our participants. It was great having you all to be a part of this event, which has helped us learn about such diverse and incredible research works. Thank you to all the participants for such brilliant work. We'll be back after a short break at 1:30 p.m. for another inspiring and ingenious session of Innovative Idea Challenge. Thank you, everyone. You can leave now. And I would like to make an announcement to join at 1:30 again after a short break.
Good afternoon, everybody. Welcome to the third day of the third Student Research Congress, organized by SVKM Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by University of Mumbai, industry partner, ACG Worldwide, and Cytex Center. I know everyone has been looking forward for this session, and why not? Innovative ideas and such creative minds coming together from all parts of the country. As it is rightly said, innovation is seeing what everybody has seen and thinking what nobody has thought. This is going to be such an inspiration for all of us. For Innovative Idea Challenge, the judges are our first judge, Dr. Pankaj Manpe sir. Sir has pursued M.Pharm in pharmaceutics and PhD in pharmaceutical sciences from SPTM's NMIMS University. He has pursued Masters in Marketing Management from Mumbai University. Currently, Sir is working in R&D Center of Microlabs Limited as Senior Vice President and in R&D sector as also leading a functional team of more than 190 members, including formulators, analysts, DQA, engineers, technology transfer team, and R&D. Sir has over 29 years of experience in R&D. Sir has filed more than 15 patents for Microlab Limited. He has more than 7 research articles published in different journals, and 3 book chapters are published as on date. Sir has worked on more than 100 bioequivalent generic products for regulated or ROW or WHO markets. He has audited sites within India as well as out of India to identify potential CMOs. Few sites are working with Microlabs Limited now to supply US market requirements. Sir has expertise in product development for regulated as well as the rest of the world and WHO market and in development of various conventional dosage forms. Besides conventional processing, Sir is having an expertise in pellets and MUPs technology, osmotic drug delivery technology, delayed release formulations, fluid bed processing, and hot melt technology. Our second judge for the session is Dr. Vasanthi Suvarna ma'am. Ma'am is currently associated as an assistant professor in Department of Pharmaceutical Chemistry and Quality Assurance in SVKM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. She has total of 17 years of teaching experience and 2 years of industrial experience. Her thrust research areas are solubility enhancement, drug delivery, analytical method development, and natural product research. She has published 40 papers in reputed journals with total impact factor of 65 and also published 9 book chapters. Ma'am has guided total 23 M.Pharm students. Talking about her professional membership, she is a life member of Indian Pharmaceutical Association and also a life member of Indian Society of Technical Education. Before starting the session, I welcome all the judges.
Hello, everyone, all judges. Thank you very much. This is Munira. So thank you, Dr. Pankaj. Thank you very much. We really appreciate your time. I know that being an industry person, you are always busy, but we really appreciate your time. And I'm sure you will enjoy thoroughly the innovation entries, innovation challenge entries, and your inputs would be very, very useful to students. So thank you very much.
Great. I'm also happy to attend this congress because I wanted to see how students are presenting, what are the thoughts of current generation. I should say current generation because it's a 30 years gap between me and now whoever is presenting.
Correct,
so that's why, yeah. So, and even the topics are interested. I'm very eager to know how they are presenting, what are the thoughts, and definitely it will be a good experience.
And also, student will get the industry perspective from you.
Right. Commercial and all those aspects can better.
Correct. Yeah, I was just on a round, so I just entered into faculty room and I did not want to miss welcoming you and thanking you, so I just joined from one of the faculty's computer.
Yes.
All right, so welcome to BNCP, Dr. Pankaj. Thank you very much, and thank you, Vasanthi Madam, for pitching in. Thank you.
Thank you, ma'am. A very hearty welcome to all the judges. Before starting the session, I want all the participants to kindly take note of the instructions carefully. Rename yourselves as per the allotted codes. Participants cannot reveal their names or any affiliations. Each presenting author will be given a time of 10 minutes for presentation. Follow the time limit and kindly do not exceed. Only the presenting author will be allowed to answer in the Q&A session. Each participant is requested to stay alert for their roll call. I would now like to take the permission from the judges to start the session. Sir, ma'am, can we start the session?
Yes, please go ahead. So, can we start?
Yes, please. Yes, please.
Yes. Please go ahead, Coach.
Now I request participant number IIC 01 to be ready. Should we share the screen, or you want us to share the screen?
Let them.
We can share the screen.
Okay. Please share.
Good afternoon to one and all, respected panel of judges, teachers, and all the delegates present here. Before starting off with my presentation, I would like to quote, "Health is a state of physical, mental, and social well-being that enables everyone to live productively." The concept that we will be dealing here today are advanced microencapsulation of dual drugs for the treatment of GIT infection. The content that we have covered in our presentation are introduction, rationale, experimental details, and conclusion. Kicking off the presentation by defining the word microcapsule. It is a spherical particle with a size varying between 50 nanometers to 2 millimeters, containing a core substance. The core material is the active element and the outer covering is the shell. By using microencapsulation technology, the active material can be entrapped into a shell by using various coating materials like polymer, which can be manufactured from natural or synthetic, can be made permeable, semi-permeable, or impermeable. The encapsulation efficiency of microparticles can be determined by various factors like concentration of the polymer, the solubility of the polymer in solvent, rate of solvent removal, the solubility of organic solvent in water, and so on. The basic rationale behind this project is sustained release of the drug that increases the therapeutic effect. Dual drug delivery system, and the drug molecules entrapped is immune from the external environment. A single capsule can target two drugs at two different parts of the GIT. The coating of microencapsulation can be done mainly by two processes, which are physical method and a chemical process. Air suspension coating, conservation process, pan coating, and spray drying is a physical method. Chemical processes consist of solvent evaporation and polymerization. Out of the mentioned processes, we have used pan coating as our method of preparation. This technique entails a coating composition to a moving bed of particles. While using hot air, we can increase the solvent evaporation. The appropriate solid core material in the coating pan is covered with a coating material, which is applied at solution or an atomized spray. Since solid particles larger than 600 microns in size are typically thought to be necessary for efficient coating, the particles are tumbled in a pan or a devices along with the coating material is applied gradually with respect to the microencapsulation. This method has been widely used to create controlled release beads. Experimental details. Advanced microencapsulation is being placed in hard gelatin capsule. This study mainly focuses on a dual drug delivery to different targeted sites in GIT. Drugs having low bioavailability can be released drug for prolonged period of time in stomach. This can be achieved by increasing the gastro-retention time by using low-density bioadhesive polymers. On the other hand, another drug in same capsule can be delivered to intestine by using high-density polymer. These are the various evaluation parameters that can be used for microencapsulation, stating morphology, drug content, bulk density, angle of repose, in vitro dissolution studies, bioadhesion, diffusion study, and so on. Conclusion. Two different drugs enclosed in a single capsule can be delivered to different sites of GIT. By using this method, we can achieve the aim of gastro-retention drug delivery system, mucosal drug delivery system, and targeted site drug delivery system. Further, I would like to thank BNCP College for giving us this opportunity to present ourselves and our project in front of you all. Thank you very much.
Thank you, IIC1. Now the session is open for Q&A.
Yeah, do you have any idea about booster coating?
Sorry, sir.
You have any idea about booster coating?
No,
I would simplify fluid bed processor bottom spray. Pan coating, how are you going to do this pan in the pan? Two different beds, and what is the ratio at the time of capsule filling? All these aspects.
Sir, basically we are working on this project, but we have gathered the raw materials, but we haven't proceeded with this. We will do it in next month probably because of our current examinations going on.
Yeah, okay, but what are your plans afterwards? Once the materials are available, you will be doing only pan coating, using pan coating technique?
Yes, sir. We will be using pan coating as this is handy to use, and we have pan coating in our premises as well. So.
But do you have perforated coating pans, or it's normal? Whatever picture you have shown is not a perforated coating pan for a coating of pellet or bead. So consider all these aspects before going ahead, because you are doing particle coating. It's not a routine tablet. Okay.
Yes, sir.
Routine tablet in a pan coating is that way very simple. But particle coating, you have considered very basic approach of doing pellet coating. Industry is far ahead than what you have thought of right now. So get some more literature on. You can write down fluid bed processor bottom spray. We call it as booster coating. But you can mention or you can discuss it because Associated Capsules is a partner, I believe, for this congress. You can get much more and good information on particle coating by booster technique, bottom spray, and you can develop your product, more robust product, I would say.
Thank you very much, sir.
That's all from my side.
Yes. Just tell me, how are you going to make it gastro-retentive, like the two drugs which you have, no? Yeah.
Ma'am, we are using low-density bioadhesive polymer. In
For
case, example, ma'am, we have example such as HMPC, HPMC, K15M.
Okay.
Yes, ma'am. We can use this polymer for this has both the properties as it can adhere to the mucus layer as well as it can float. It has low density as well.
So that is for release in the stomach.
Yes, ma'am. The drug will release in the stomach, sustained
And for
release.
the
Yes.
intestine? For intestine?
For the intestine, we have used high density polymer.
Hmm. So, which polymer are you going to use it?
Ma'am, Utrigit S is one of the polymer that has high density, so we can use that.
So, what is the lifetime of the product in the stomach? How much time it will be having resident time in the stomach?
Ma'am, sorry, we haven't processed further.
Otherwise,
But,
for formulation, you must have done literature survey, right? For
yes,
the
ma'am.
gastroretentive formulations which are available in the literature, what is the residence time in the stomach?
Sorry, ma'am, I'm not recalling it right now.
Okay, and which type of combinations? Are there any product in the market for the concept which you have proposed?
For this particular concept, ma'am, there is a none product available in the market right now. It's we are doing for first time. I have thought of this project. It's not available. There are various.
Yeah.
Sorry, ma'am.
Microspheres, you meant to say, but there are floating formulations, no?
Ma'am, there are floating formulations, but we are using a combination of floating as well as high density to target two different impacted areas. One is that our stomach, and second is inflammation in the colons.
So you meant to say the udrated polymer coated layer is not going to be released in the stomach. That's what you're saying?
No, ma'am. We we are using pH sensitive polymer, so it cannot like.
Which is pH sensitive polymer? Udrated.
Yes.
HPMC. Yeah.
Utrigit S.
You're going to coat the solution on the microsphere. That's what I've understood, right? Or you're going to incorporate your drug in the microsphere, or you're going to coat it on the microsphere?
We are entrapping the drug molecule. We are in the microsphere. We will be.
So your polymer is not going to be released in the stomach, the one which you are targeting for intestine. Correct? That's what you're saying.
Yes, ma'am.
Okay. Okay, fine. That is it from my side.
Thank you very much, ma'am.
Thank you.
Thank you, IIC1. Thank you, judges. Now I request IIC2 to be ready. Will you share the presentation, or you want us to share the presentation?
I'll share the presentation.
Okay, okay.
Is it visible?
Yes, it's visible.
Okay. Hello, greetings. Hello, greetings. So the topic is Savdhan Wearable Devices, an amalgamation of healthcare and artificial intelligence. What are difficulties encountered by a specially abled person? Number one, problems in identifying and navigating the routes.
I cannot. Hello, the presentation is not visible to me. Can others see this presentation?
Sir,
At least I cannot.
It is visible. I think there is little lag. Sir, shall we wait for some time?
Yeah, please.
Yeah, it's visible actually.
Shall I continue?
Just a moment.
Okay.
Just a moment, wait. Sir, can you see now?
No, I can't.
Kunj, are you able to see?
Yes, yes. There might be a internet problem from the sir's side.
Sir, if you could check the.
Yeah, now it is visible. Yes.
Okay,
yes.
sir.
Okay.
Yes, candidate, you can go ahead.
Shall we start again?
No. From the first slide itself. No, she's in the first slide only. No?
Okay.
Yeah, start.
What are the difficulties encountered by a specially abled person? Problems in identifying and navigating routes, potential risk for accidents, unmodified floors and sidewalks, and inappropriate obstacles and hurdles can increase the chances of accidents. What would be the tagline for our product? Better senses, better life. And this will be the logo for the same. The vision statement is to provide novel wearable accessories at an affordable rate and thus enhance the quality of specially abled person. And the mission statement is to aid specially abled people to live a comfortable life. Let us have a brief background. WHO emphasis on the resolution of problems that are associated with visual impairment. The development and implementation of eye care services is a need of an hour. And thus, this is one of the eye care services assessment tool. The problem statement is sensing of the nearby hospital and difficulties in the navigation of the routes by specially abled people. And the proposed solution for the same are ultrasonic distance sensors for navigation and efficient commutation with Arduino Mega technology. Number 2 is designing wireless medical patches with the same technology for navigation programming. And number 3 is bringing out and introducing different wearable accessories like purses, wallets, gloves, shoes, instilled with this Arduino Mega sensor. So these are ultrasonic sensors that are used for navigation and efficient communication. It has an ability to sense up to 10 meters of distance as it emits an ultrasound of 40,000 hertz and 40 kilohertz. For wireless medical patches, we need a microfluidic chip and an integrator. How would be a small mechanism of action? Simply just adhere the patch with the Arduino tracker installed within it, and the data will be automatically sensed due to pre-feed softwaring, and then the desired action can be taken. Now, how does the person come to know whether he has to sit, jump, forward, what action is to be taken? So once a specially abled person comes to know that there's a device there's any obstacle in the nearby vicinity, there would be different alarms and different sounds produced by this wearable device, and thus it will help them to know what action is to be taken. What would be the target market for these devices? India is the home to the third of the world's most blind population. The country has around 12 million individuals with visual impairment, and in the global of 39 million. There are around 17.3 million people with visual impairment in China. So the Asian market would be the biggest market for marketing our products. What would be the strategies? Selling these products for blind age homes, eye hospitals, NGOs, and Make in India. Tie-ups with the Make in India for revenue generation. How is it different from the other blind aids that are already existing in the market? Others will be selling at around 2,600. This device will be waterproof. This device would be made from electronic waste, so the recycling of the waste is also possible. The packaging would be environment friendly, and the others might have Arduino Nano or Uno technology, but this device has Arduino Mega technology. That is a peculiar feature of this device. And these are the competitors that provide similar products: Orcam, Lumen, Tactile, and Iris Vision. So this is a brief block diagram that how would the device exactly work? So if a specially abled person is wearing this device, he would come to know the nearby any vehicle, any obstacle, and thus there would be alarm and notified by the person. This would be the product design and the budget at a large scale level. The smartwatches integrated with the Mega sensor, Arduino Mega sensor, would make a final product around 1,500 rupees. And what would be the future scope of these devices? We can either make an application that is AI-based application, or amalgamating these devices with a highly sensitive critical technique. We can also design various novel wearable devices, because a person might feel a little embarrassed when he's carrying a stick and checking out for the navigation. So these devices can also reduce that embarrassment and help the specially abled people to feel like a normal person only. They wouldn't be different from others. So this would be the future scope of the device, and it is also aiding in NFC, that is near field communication, that is very vital for specially abled person. What are the different revenue generation programs? Can be used for palliative care patients. Online marketing can be possible of the same. Patient with poor vision, either suffering from glaucoma or any other iris disorders, tracking the device for the patients that have dementia, pre-senile dementia like Alzheimer, or any psychological problem, and then a special complimentary gift for the same so that marketing can be enhanced. What would be the SWOT analysis? Strengths of the product are highly sensitive, capability to capture a wide market, and bringing out large number of wearable accessory devices for a person. What would be the weaknesses? Soon the 3D bar printing might hamper the growth, and the Arduino sensor has a shorter lifespan, so it requires a quite frequent replacement. What would be the opportunities? According to WHO, 253 million people are living with visual impairment globally. So it can help in capturing the global market. And the threats are some other company might manufacture these products with our new technology, upcoming technology in future. These are few of the references that I've adopted for making this presentation. Thank you.
Thank you, IIC2. The session is now open for Q&A.
Yeah, it's a wonderful presentation. IIC2, I would say. I have a few questions. When you said the applications, you will approach hospitals and all. What about training?
What about the training? Yes, sir. So the training programs we can actually bring out, just like there are leaflets and there are few informative, we can, whenever we buy any balm, suppose any other balm, so there is leaflet inserted. So we can provide a leaflet for the same. And if the people are not literate, if they are not able to understand the language or something, then we can also get some few volunteers so that they can demonstrate and they can perform live demonstration and give training to those people.
Okay, why you have selected a smartwatch?
Sir, because number one thing, there are many sticks available, but whenever we see when a blind person walks with a stick, he might feel a little inferior, he might feel a little embarrassed while walking. But when he's wearing a watch, he has a different confidence, and it is also very compatible. You can directly wear it on the wrist. You don't have to carry, it's not so bulky or it's not too difficult to carry it somewhere. So that would be an advantage of the watch.
Right. Your target population, if you say it's India, then in general, if you see the whatever 12 million population which is blind right now, the classification with respect to income group in this total blind population, have you given any thought for that? Again, I would say in general, we have a sympathy or mentality towards blind people who are walking with using the sticks. Okay, so in that case, suppose if someone is traveling or crossing a road with having this typical red stick, definitely there will be some sympathy towards him. But if he's wearing watch, how will people identify that he is a blind person? And so my next question would be a pothole at very short distance.
Yes, sir. So,
considering the bad situations of road and all, so a pothole at very near position, how we can
even need anyone's help. He might himself know, suppose if there is a pothole nearby, so that sound, there would be a different beep sound. So he would come to know, there's a pot ahead, so I need to walk from left and right. And similarly, there are different alarms and there are different sounds. So that sound would help him to know what action he has to take, whether he has to jump, sit, walk left or walk right. And the second question that you told about the potholes that are very close by and very nearby, how would it detect? So as I mentioned, again, it can detect up to 10 meters. So even if it is very close vicinity even if it's very close, here you can see it can detect up to 10 meters. So yes, the nearby potholes will also be detected by that watch and any novel wearable device associated.
And which battery you are using for this smartwatch? Rechargeable battery?
Yes, sir. The batteries would be rechargeable, but yes, again, it would need frequent replacement, as I mentioned, because the sensor has a has a lifespan not greater than a few years. So yes, replaceable batteries would be used.
A blind person alone at home, can he do this recharging of smartwatch and all? And what will be the battery life? After how many hours he has to recharge once it is in regular use?
Sir, he might, of course, that he might need assistance because he will not be able to himself recharge the battery. So he would need assistance, someone's assistance for that.
So thank you, IIC 2.
Thank you.
Thank you, judges. Now I request IIC 3 to be ready. IIC 3, are you there?
Yes, I am here. Are you able to see me and hear me?
Yeah, should we present the screen or will you present the screen?
No, I'm presenting. Just give me.
Okay.
Is my screen visible?
Yes, it is visible.
Can I start?
Yeah, your time starts now.
Okay. So, good afternoon, one and all present here. This is IIC 3 and today I'm here to talk about the production of bioelectricity from industrial waste using microbial fuel cell. So basically what the problem which I'm here dealing with is like, first of all, like according to a UN report, 80 to 90% of the wastewater in developing countries is discharged directly to the surface water bodies. So what happens when we discharge this wastewater into the surface water bodies? The aquatic environment inside this surface bodies, like fishes and all the creatures which are living, the lives get affected. Addition to that, all this contamination which is caused, and this water is then consumed by the people living around, and people this water used for human consumption. So in this case, due to this, there are various diseases which are caused like cancer, diarrhea, and various other digestion problems. So this is the first problem. Second problem which we are facing in the current world is that our dependence on the non-renewable energy resources is keep on increasing day by day. As we can see in this graph, like although like 10% of like from 2008 to 2018, our dependence on the renewable energy has been increased, but the non-renewable energy resources have significant amount of still have like 75% we are dependent on the non-renewable resources for our energy consumption. So currently world is facing this energy crisis also, and this wastewater is directly dumped in the surface water bodies. That is also one problem. So this is my problem which I'm facing. I'm dealing with today. So this is you can see in the upper right corner there is a Tom cartoon is there. So like another Tom from future is coming and saying him that please save me bro, and from extinction that I am going to die, so please save me. So like it is truly said that we have not inherited this world from our ancestors. We have borrowed it from our future generation and we have to return them safely. So what innovation and what the solution which I'm bringing today is, it is a microbial fuel cell. So it is a one of a kind of technology which does bioremediation of the waste and produces electricity from the from this waste simultaneously using eco-friendly microbes. So energy produced by this technology is very sustainable than other forms of energy. Like I would like to compare this energy source with other forms of energy. Like if you take an example of hydroelectrical energy, it produces energy only once in a year when the dam gets filled with the water. Tidal energy, you can only have it during the high tide time. Solar energy, it only produces during the daytime. Then is a thermal energy which causes air pollution and air pollution all very much. Then there is a nuclear energy which is very costly and very hazardous. If we look into the this technology, it uses microbes. So like microbes, when they consume this waste, they grow their population and energy keeps on increasing day by day. And whether it's a day or night, it produces the energy. Then if this energy is commercialized, it will be very a revolutionary step towards saving the environment, and green energy would be produced. So like now I will explain the working of this reactor. So basically here we have a two compartment is this. Basically the microbial fuel cell are of two types, single chamber and the double chamber. So for industrial purpose we use this double chamber model. So in the first chamber it is an anode chamber, and in this anode chamber we will incorporate our waste and we will incorporate our microbial culture, and the anode electrode which would be there, it would be rich in the carbon so that the microbes can surround around it and can transfer the electrons. And second component which we have here is it is a membrane. It is a membrane for the transfer of the protons to the cathode chamber. Cathode chamber will have a normal electrode, and it would only fill with the water so that it has the access to the oxygen so that the reaction could be complete, and the circuit would be completed, and in the end the charge would be the electricity will produce. So basically what our input into this reactor would be waste matter and microbial culture, and our output would be water and electricity. So like when we upscale this model, the basic like problem which we face, like the transfer of protons from anode chamber to cathode chamber decreases because the space between the two electrodes increases. So to overcome this problem, we can incorporate the chemical mediators which will mediate this electrons and which will mediate these protons towards their destination. So moving forward, like what would be the business model of this technology? So like first of all, vision, we need to stick with the vision, like we need to produce the better world for tomorrow. So and then second would be messaging, like whoever would be our client we should be able to understand, like make them understand the problem which we are facing and how this technology could help them. And then we also need to have agreement with them. Then we need to keep the cost as effective as possible so that the maximum amount is maximum effectiveness is achieved. Then like we need to like also go for the marketing sides of this technology, because the awareness about this technology is very less. Then there are various growth opportunities, because I will discuss this in the next slide. There are more growth opportunities. Moving forward, like what would be the market and the target audience? So this technology can be implemented in all those industries which discharge a toxic wastewater in water bodies, for instance, like petrochemical industries, food processing industries, pharmaceutical industries, electrochemical industries. And this technology could also be beneficial for economical backward countries, as it is very cost effective. I remember the last time I was researching on this technology, one of the individual was charging his mobile use of this technology in Africa. So like we can use this technology very effectively. And one more thing, like we hear very frequently, like industry want to achieve their zero waste goal. They want to become more eco-friendly. So this technology will make industries more sustainable towards the environment and will help them achieving that zero waste goal. So what would be the future roadmap of this technology? Currently, this technology is in prototype phase. For further moving forward, we need to collab with the industry. We need to upscale this prototype. Then we need to conduct the trials and what waste we are incorporating, how much waste we need to incorporate so that what amount of electricity is being produced. Then we need to approach other target industries, and like let's say if I mean being very much superficial, like by 2030 we can see this technology in 50% of industries. So these are the references which I used in my presentation, and I also have the prototype of this technology. If jury allow me, can I show this prototype to you?
Yes.
Okay.
Yes.
Yeah. So, hello sir, hello ma'am. So this is the prototype of this technology. I have made a single chamber microbial fuel cell. So are you able to see this beaker?
Yes.
So basically in this bottom layer, I've incorporated the waste and the microbial culture, and I have also incorporated my anode electrode in this. So what is my anode electrode? As I said in my presentation, I want to have a carbon rich electrode so that my microbes can grow around this electrode. So I've selected a thing called char cloth, which is basically a cloth burned in the absence of oxygen so that it becomes rich in the carbon content, and I've wrapped it under the aluminum foil with the back. And second, if you come upwards, there is a layer of don't know if you can see this. Like it's a agar, salt and water gel. Meet this gel and freeze it. You can see layers here. Like it is a circular waste in my this microbial culture, and anode is inside it. Layer is there, and above it is the water. As I said, in the water. So water is there. Then these are the wires, and this is a multimeter through which I will amount of electricity is being produced by this prototype. So I will
only 1 minute is left.
And the yeah. Thank you for the charge is being produced, and to be precise, around 330 to 400 volts is being millivolts is being produced currently at this time from this prototype presentation. And this I don't think that this prototype and this technology in every and thank you BNCP for this wonderful opportunity. Yeah.
Thank you. Now the session is available for Q&A.
Sir, please go ahead.
Yeah. What do you think the quantity of waste required for suppose a day's consumption?
Sorry, sir, I'm not
type of waste or
again
quantity.
Quantity of the waste.
How much waste is required?
Okay. So
if any individual
so basically
need to complete this bioanalysis.
Sorry.
Any pharma company involved in making this type of micro-based fluidic cell? Fuel cells, sir is asking that. Any company which is marketing this type of microorganism based fuel cell?
No, I don't think is commercialized. As you sir mentioned about the what amount of waste or what kind of waste. So basically, we create this waste which we incorporate this so that energy would be dependent on the waste has. So I read some research papers. If your waste has 2000, will produce around 97 watts of electricity of power. So that sir, that energy calories your waste has.
And you have shown this agar plate, right? If I'm not wrong, you have used agar. So how about disposal of this agar?
Sir, we have this thing in our institution, so we will be generally go for it for this decomposition of this agar, or we can bio waste like our
Again, then it will not be a burden if you say that it is a bio waste. Will it not be a burden? Because bio waste needs special treatment. Even we normally we don't send our waste. We have a tie-up with one company or organization who are dealing with whatever waste we are generating in the R&D center. So what do you think this bio waste? See, in the process of making electricity, okay, you are generating some
waste.
Bio waste. So you should give a thought on this disposal, and it is not easy to dispose all these agar plates and everything. So more and more technology means much more technology is available in the industry to avoid agar plates. I'm saying, and if your basic work is your basic work is based on these agar plates and all, destruction of these, how it will be generated in the premises where you are putting this plant. All these factors you have to consider.
Sir, this was just a prototype for agar. If we upscale this model, we would not use agar. We use polymer membranes for it. So agar would not be used in the industrial model.
Right, but then scalability, because you are working on a model with using agar as a base or whatever starting ingredient, and then for the industry you are thinking of some polymer. So what is the scalability of this project?
Sir, scalability, sir, still as I said that upscaling is remaining of this, and we upscale this. There, as I said, technology has been incorporated into the microbial cultures. So like still much work is to be done for the biotechnology side, and like this chemical mediator is already being done. So like still such scalability issue is faced because the lack of research in this aspect, because this prototype is very multi-dimensional, because physics is also involved, chemistry is also involved, and like biology is, everything is involved. So like we need to get the people from all the professions on the table and work on this project.
Fine, I'm done, ma'am.
Okay. I just wanted to ask, you are treating the bio waste, right? Organic waste. So at the end of the process, is that waste is in a position to be disposed in the environment as such, or that bioremediation you're doing, right? So the organic waste that you'll be using for generation of electricity, how will it be? Like, will it be environment friendly at the end of the process?
Yes, ma'am. That was the basic concept. Like we need we do the bioremediation of the waste into this technology, like microbes. What is the waste for us? It is a food for microbes. So microbes like decatalyze those materials, so they are completely harmless after going from this technology.
And microorganisms that you'll be using for the process, right? If are they environment friendly, or like they are causing some infection? Which type of microorganisms are used?
So microbes class which are being used, it is basically the geobacter. Geobacter class of microbes. They are very environment friendly. Like you can find them in soils, and like they are very eco-friendly microbes.
And where you are going to set up this plant, as sir said, no? Will there be any gas generation or something in the plant? Like, do you require an enclosed assembly, or it will be an open? Because a lot of microbiological degradation is going to go on there, right? So is there a gas generation or the byproduct of this cell, micro fuel cell?
Ma'am, as I said, our input is waste and this microbial culture, and output would be only H2O, and this H2O would be there and water would be there. So as such, there would be no gaseous emission from this cell, from this reactor.
Okay, so as you said, carbon rich waste it has to be. So is there a requirement?
Carbon rich waste, electrode needs to be carbon rich.
Electrodes need to be carbon rich. But use the domestic waste, can that be used here?
Yes, ma'am. One researcher named Bruce E. Logan is there. He has dedicated his life to this microbial fuel cell, and he has worked on different type of waste, domestic waste also, industrial waste also, and he has found out that domestic waste can also be used for we can be like incorporated into this, and like they can also be like decomposed using this technology.
So how do you rate biodiesel or the gobar class gas plant which we have over this technology, MFC, that you are proposing? In terms of costing and the output, the investment.
If you talk about biodiesel or biogas, they are accepted into our society. Like people use that, they dump their cow dungs into it, and the energy is being produced. But this technology is completely different. It uses the microbes, and it is a more of a like industrial purpose. it's there. If you use the domestic waste, it may not be able to generate that amount of energy, or so that if you incorporate this, biogas or biodiesel are still not been incorporated into the chemical industries. So this model is very much suitable for the industrial purpose.
Okay, and what is the limitation? What is the highest power density that is obtained by this MFCs?
Ma'am, as I said, the power generation completely depends on the what amount of waste you are incorporating. So if you incorporate the glucose microbes will
So in the literature, can you comment on because you must have done a lot of literature survey, right? So what is the highest amount of electricity and the corresponding waste for that?
Okay, ma'am, highest amount of energy which was produced, as I said, from 2000 calories, it was 97 watts, which was around 100 watts, you can say. And the full bioremediation of that waste was done. And like they had conducted trials of toxicity and studies after incorporating those waste into this. So like they were completely bioremediated.
So any company who is marketing this type of cell?
No, ma'am, there is no as such because this technology is still under the research and it is not yet commercialized, ma'am.
What would be the probable reason? Because biogas we have in place now in our ecosystem. So what is the reason for this not being commercialized yet?
And the basic, as I said, it is a very multidisciplinary thing. So like you need to get the people from all the profession on the table, sit on that, because there is a physics involved, there is a chemistry involved, there is a biology involved, there is microbiology, chemistry, everything is there. So like basic reason is that extensive research is required on this. So that's why it is not yet been commercialized, but it has a huge potential because many research paper and many small scale models are being made of this technology.
Okay, thank you.
Thank you, ma'am.
You IIC3. Thank you, judges. I request IIC4 to be ready. IIC4, do you want us to share the presentation or will you share the presentation?
We'll be sharing the presentation.
Okay. Okay. Your time starts. Now.
Okay. So, good afternoon and welcome to one of our present here. We are presenting our topic for the Innovative Idea Challenge, which is the Whey Lemonade, a potential midday meal supplement. Firstly, we shall talk about the importance of protein and its role in daily function. Protein is one of the three big macronutrients. It is extremely important in children and adolescent individuals to ensure proper development. It is needed for the production of hormones, enzymes, hemoglobin in blood, skin, hair, and nails. Now we shall talk about protein deficiency in India. India as a culture is very carbohydrate heavy. We usually prefer to have carbohydrate heavy meals, and therefore most of us are protein deficient. The Indian Market Research Bureau had a report in 2017 which stated that over 80% of Indians are protein deficient. The Indian Medical Bureau suggests that Indian adults should consume close to 0.8 grams to 1 gram of protein per day per kg of body weight. However, Indians usually consume closer to 0.6. Also, Indians spend a very small amount of their food budget on protein-rich meals. They usually spend less than 1/3, with most of the money going towards processed foods, which are heavy in carbs, sugars, salts. Now moving on to the impacts of insufficiency of protein on the human body. Now, a lack of protein in the human body can cause a variety of diseases, from mild cases like weakness and fatigue to more concerning conditions like muscle atrophy, liver failure, and stunted growth. In children and pubescent adults, it can lead to a myriad of growth and developmental issues which will stick with them for life. Protein is important for extremely simple functions in the human body, like retention of water in blood vessels, which is controlled by the presence of albumin. The failure of this albumin to be present in the body leads to the loss of water from blood vessels, which causes bloating and swelling in parts of the body. That's a very key symptom of kwashiorkor, which occurs in a lot of poor and underdeveloped countries. Lack of protein is also the leading cause of death in young children in these poor and underdeveloped countries. Now, this is the Global Hunger Index, also referred to as the GHI. It is a tool that was used to track and measure hunger at any level. So, in this case, it's on the national level. It was developed by Concern Worldwide and Welt Hunger Hilfe, which is a German organization. So according to this, it is visible that India, out of the 116 countries that were measured, India has a rank of 101 with a GHI of 27.5. That is considered to be in the range of serious. Now coming to the protein requirement for children, children are growing humans, so they require a lot of protein for muscle, for brain, and all of that. So this table tells us the reported protein requirement for children ages 1 through 13. The grams per day and grams per kg per day values are all on statistical analysis of the heights and weights of most of the children in this age group.
Okay, so whey can actually be used as a source of protein. Whey accounts for about 85 to 90% of the volume of milk and contains about 55% of the nutrients in milk. Whey is obtained from milk as a byproduct during paneer production. The process for making paneer is well known. It is a process that involves boiling milk and curdling it with food acid like lemon or vinegar. The lumps formed are called curd. This curdled milk is then strained and pressed in a muslin cloth, and the residual liquid is called whey water. Whey contains 0.7 to 0.8 grams of protein per 100 ml. So 1 cup of whey water contains 2.1 gram of protein. Additionally, whey water also contains vitamins and minerals like calcium, iron, magnesium, and also vitamin C. This whey water can be refrigerated and preserved for about 5 to 6 days. The environmental effects of dumping whey. As we know that the dairy industry produces huge amounts of whey as a byproduct. Large-scale industries have already adapted and introduced new generation products like whey protein powders, beverages, etc. that utilize this whey water that they produce. This minimizes the amount of waste generated. The small-scale dairies, however, they don't use the whey water that is generated and they simply discard it. Our idea is about whey utilization and developing a sustainable use. Dumping waste significantly increases the BOD due to the high amount of organic matter present in it. Lactose, fat, and protein constitute the main fraction of the organic load. Disposal of whey represents a significant loss of potential nutrients and energy. So, in order to utilize the nutritional value of whey and at the same time reduce the harmful effects it causes, it is very important to direct whey management towards a cost-effective and sustainable way of utilization. The midday meal in India. The midday meal scheme was launched in India on 15th August 1995. It was aimed for classes 1 to 8, that is, children between 6 to 14 years of age. The midday meal is a freshly cooked lunch that is served to children in government and government-aided schools in India. The objectives of the midday meal issued by the government is to improve the nutritional status of children, encourage children to attend school more, and concentrate in classroom activities, and also address malnutrition. As midday meals don't entirely fulfill the protein requirement needed for children, our idea is about providing a midday meal supplement that can improve the protein content to some extent. You all have now been led to the idea that came across our minds when we joined the dots between all the pertaining issues, and that is
we eliminate a midday meal supplement. Well, let's talk about the details. Now, 1 kg paneer is made with 6.5 to 7 liters of cow's milk. Small-scale dairies usually produce about 20 kgs of paneer on a daily basis by using close to 140 liters of milk. We came upon one such small dairy in Santa Cruz that discards huge amounts of whey water that is formed after the production of paneer and was also willing to let us take as much as we needed free of cost. We realized that these small dairies are unaware about the potential uses of whey. We can obtain close to 115 liters of whey water from a single small-scale dairy in a day. The ingredients for the recipe include lemons, sugar, black salt, and whey water instead of regular water. The rates of each have been mentioned on the slide and have been taken from IndiaMart to avoid regional differences. Additional agents like fruit extracts and jaljeera powder can be added for flavor. Basil seeds and mint leaves are an excellent source of both nutrition and flavor. Fruit pulps are a natural sweetener, along with being a source of protein, fiber, and loads of nutrients. It's important to take a note of these agents because the likability of the children for the drink is directly proportional to their attendance. These agents have multiple health benefits, and of course, the sweeter, the better. For example, talking about Japan, the midday meals are well-balanced, tasty, and nutritious. This directly results into greater attendance and more the number of people attending school, better the work ethics, and ultimately a highly developed economy, being the third largest in the world. A survey was conducted in September 2022 at SDSOS and MIMS to check how people responded to the idea and quality of whey water. A total of 20 responses were collected, out of which 65% of the people rated it 8 and above out of 10. Whey water was collected from a local dairy in Santa Cruz. 1.6 liters of whey water, 3 lemons, 4 tablespoons sugar, 2 teaspoons black salt, and 2 teaspoons jeera powder were added. These are the pictures of the process. The link for the survey has been added. The questions included were about whether it is appealing or tempting, rating the taste, texture, appearance, overall rating, and satisfaction. People were also asked to add comments or suggestions, if any. The following results were obtained for a rating of 8 and above. Taste, 66.7% of the people liked it. Texture, 62%. Appearance, 55%. Satisfaction and overall rating, 73% of the people were in the favor of it. We have now successfully addressed some of the major issues, including a solution to improve the protein content of the midday meals. The addition of whey lemonade to meals results in the improvement of the global hunger index, and the environmental impacts caused by whey water dumping will be reduced to a significant amount. But most importantly, we have made everyone's favorite drink, the lemonade, healthier. This is the bibliography.
IIC4, your time
Thank
is
you
up.
Thank you for your time and patience. We hope our idea is the way forward with nutrition. We're looking forward to any questions you all might have.
The session is open for Q&A.
Yeah, it's a wonderful presentation by you all. First of all, my few basic questions are like, will there be any control on usage of this whey lemonade? Because in that case, suppose 3 or 4 bottles or cups are consumed, what will be the side effect of this extra protein, I should say, for the body?
First of
Have
all,
you given any thought on that?
So first of all, we never expected to reach that level because this will be measured out and given to the children. We plan to partner up with certain organizations like Akshay Patra and maybe get grants from trusts like Kusuma Trust, which and work with other small organizations that feed children daily. So we never expected to reach 3 liters or 4 liters of this whey water like reaching a child's body. But if it does, the small things we can expect are stuff like damage to kidneys, proteinuria, stuff like that. But we never expected to reach that amount. It will be a specific amount given to the children based on their age group.
So some instructions are needed. Okay, you work on that while consuming this whey lemonade. Then I would say the taste trials which you have conducted are with your friends or fellow students, which according to me is not the class which you are targeting.
Yeah.
So what is the reason?
We did plan to do a taste with certain organizations or even with medical institutes that
No,
have, like,
sorry.
take care of children.
Sorry to interrupt you. I'm talking about when you said you are planning to do with Akshay Patra and all for midday meals, then that class of students is entirely different.
Yeah.
And the product which you have tested in the class is a totally different class.
Yes.
So whatever test trial you have conducted, there is a possibility might be the students have compared the taste of existing lemonade what they are drinking daily against the way lemonade, and they must have given you feedback. So give a thought on that part as well before proceeding. And my last question is on usage of sugar. Why you have made it with sugar? Why it cannot be made a sugar-free lemonade? Because there is a possibility of diabetic patient, even a child by birth. So in that case, due to unawareness of all these issues, like consumption of sugar, or if he is diabetic, he or she, a kid, then the impact will be severe.
Yes, sir.
Because
That's the
protein plus
reason.
you are giving a sugar.
That's the reason we have added additional flavoring agents. So, like, if we use a fruit pulp extract, then it's healthy also, and it's like sweet also. So the taste will be there. We don't necessarily have to use sugar, but since like sugar is cheaper, so we can use that.
Avoid sugar. That's the thing. Don't go by cheapness or whatever the cost. If you are targeting a product, a healthy product for as a midday meal supplement, then don't think about all this. Why? Because sugar is available cheaply and all. Don't think. Then you are deviating from your main focus. Okay, because everything you have worked out so well. Then don't deviate by adding this sugar and all. Taste may not matter if a child is seriously in need of proteins. Taste may not matter. And as a fact, children are basically, you must have observed, they can consume any bitter medicine very easily, a liquid, syrup. They don't find anything wrong in consuming all that. So taste may not matter in this case. If you want to add sugar just to enhance the taste and all, you target your focus. Focused attention is most important. And you have to indicate how much whey is present per serving. Have you calculated that?
Yeah, we plan to give a serving of 200 to 250 ml depending on the age group, and that will consist close to 1.8 to 2.2 grams of protein.
Okay, fine. Madam, please.
Yes. In this lemonade, no, you are having the fruit extracts. Okay, what is the compatibility of that with the whey protein that will be present in the whey water? Is there be some compatibility issue?
Compatibility, as in the fruit extract is purely for flavoring purposes. There is no nutritional value per se that is added by the flavoring agent.
So will there be any precipitation of the protein or something that you observed? It's a clear solution.
No, okay. So the one we took from the small scale dairy, so that we don't plan to use that exact liquid for the consumption of children. There will be requirement of a small amount of treatment to it because there is a small amount of fat present. Because after a while, there is a small amount of precipitation that occurs. So all we need to do is remove that small amount of fat, and after that, the protein doesn't precipitate out. We just add the flavoring agent, the salt, and then we can serve it.
Because additionally you are adding salt also, so there is no precipitation.
No, there was no precipitation when we did the survey. The only precipitation occurred after like 1 hour, 1.5 hours, when there was a small amount of paneer that could be seen on top due to the fat content. Yeah, that's all that we saw.
It's unprocessed preparation, right? So
the particular survey taste test we did was unprocessed. However, we do believe there will be requirement of small amount of processing to remove the extra amount of fat.
So are you planning to add some preservatives or something? Or how are you going to take care of the shelf life of it?
The shelf life, as it was already discussed, is close to 5 to 6 days. We don't plan to like make it keep it, and then hold on to it for weeks. The plan is to like make it and then in a day or two have it distributed out to the schools.
Transported under in cold storage or how?
Yeah, so it will have to be in cold storage, but our previous idea was actually ice pops. So, but then we moved from that to lemonade because that would require greater amount of energy for transportation and therefore greater cost and all. This preserves the nutritional point of it along with the lower cost.
How about the microbial load on in the preparation? Because okay, while making paneer, it is heated. The milk is heated, and then you
The
make
milk
paneer.
is heated, and then the plan is to directly take it from there and put it in cold storage, but it doesn't have to be freezing cold. And then in a day or two, it's
Take care of
given
the
out.
contamination part.
Yes,
There
yeah,
should
the
be
contamination.
because it is unprocessed and without heating. After that, you're not eating it.
Yeah. No.
Yeah. So the kids have to consume. So with the low immunity, that could be one issue. So that you have to take care.
We need to work with big, well-known NGOs who know what they're doing and can provide the logistical.
And as Sir said, no, serving is very important because
Yeah.
in midday meals or the kids in the schools, BMC schools and all, they're provided with milk. Okay, so compared to milk, your whey protein pros and cons you have to see because milk is going to contain the protein and the casein part. So it's a complete nutrition. So though whey protein is in boom, okay, there could be like some health issues that are coming up now, as you said, related to kidneys and all, excess of protein, because milk is the balanced fluid for kids. Okay, so that part serving is very important. You got to optimize based on the age group, the requirement of the protein, that is important, and compatibility, as I said. And third thing is sugar, as Sir said, no, it might lead to weight gain in the kids. As such, nowadays kids more relying on the junk foods. Okay, so if you additionally add sugar and you are claiming it as a health drink, then it might lead to the weight gain in the kids. So you got to optimize on the sugar part. You might prevent it. That's better. That's from my side. Thank you.
Thank you.
Thank you, IIC 4. Thank you, judges. Now I request IIC 5 to be ready. IIC 5, are you ready?
Hello.
Yes, we can hear
Am I
you.
audible?
We can't see you. Yeah. Will
Am I
you share
visible?
the screen or should we share the presentation?
Your screen
Yeah,
is
you're
not
visible.
visible.
I'm going to present.
Okay, please.
My presentation is visible.
Yes, it is visible.
So, good afternoon, one and all present. This is abstract code IIC 05. Our concept for today's innovative idea challenge is a smart jewelry, which can be the future of health monitoring. These are some existing diagnostic medical imaging systems. These images are utilized by healthcare professionals to evaluate and assist in diagnosis of a disease state. X-ray, MRI scan, CT scan, PET images, they uses the radiating imaging system. And whereas ultrasound, it uses sound waves to generate the images. So there are certain drawbacks of the radiation's medical imaging system. As we all know that the patient has to lie in a supine position and high doses of the radiation could cause a loss of hair, damaged brain, thyroid gland. And the process is relatively expensive. It for the long-term usage can cause the allergic reaction and also the report might take a long time. So the radiation exposure from the medical imaging is possibility of acquiring cancer. So this is our proposed smart jewelry, which is a wearable ultrasonic medical imaging device. So we know that traditional imaging system, it requires a bulky and specialized equipment, which is available only in hospitals and doctor's office. So our proposed smart jewelry is a new design which might make the technology as a wearable and accessible as wearing conventional jewelry. It is a tiny form of transducer that when activated create a sound waves that are eventually translated into electrical signals and images that may be viewed later in a mobile application. It is nothing but miniaturized form of the imaging processing system. So why we are going for ultrasound imaging? Because it gives a better look at your soft tissue diagnostic imaging, no ionizing radiation exposure associated with the ultrasound imaging. And also it is a cost effective and accessible. Ultrasound is non-invasive and doesn't require any poking or prodding. Early detection of disease, example prenatal disease, can be diagnosed. And also it is not limited to a particular disease. It's a wearable jewelry, so we can use it for the purpose of many diagnostic purpose, such as to view to diagnose the thyroid gland. We can wear necklace. So these are the advances in the ultrasound imaging system, as we can see in the first picture, such as improvement in machine. So the machine size is reduced and the screen size is increased. In the butterfly IQ device, we can see the picture is viewed through the phone rather than in the big screen. And third is ultrasound adhesive, which is nothing but a sticker in which a chip is inculcated. Third, now conventional jewelry. We all know that from the ancient civilization, which is appreciated for the jewelry, and it was used to highlight the natural beauty, and different pieces were worn to symbolize the different messages such as security, wisdom, and elegance. So, what is the rationale for smart jewelry? Smart jewelry refers to the variety of rings, bracelets, necklaces, and earrings that fall under the umbrella of wearing technology. As I said earlier, that many kinds of rings, bracelets, and necklaces are used so that, for example, we can use a headband to diagnose any kind of a disorder such as Parkinson or Alzheimer. It aims to improve the health, communications, and productivity in our life, addresses the smart medical sensing in elderly and children with a special need, especially in a manner that is convenient and simple. This information may be remotely transferred to a smart device, enabling the caregivers to deliver the proper medical care as needed. So here from my teammate will continue.
So here is the proposed smart jewelry model. As you can see, it consists of a smart jewelry design. It can be available in various forms, like as previously my teammate has said, like it can be a headband or it can be a pendulum, anything. The next is the slot, which consists of the transducer chip, a long-lasting coin cell, and below which is a metal plate. The special feature of our device is that this slot is removable. So whenever it is not in need, this slot can be removed and it can be worn as an ordinary jewelry. So the working of this smart jewelry is that like if we want to capture the image of a particular part, so we just need to apply the gel at that particular part, and this jewelry first we need to put in the slot, and then we need to move this particular jewelry in a circular motion in over the part, and the image which is obtained will be viewed in the smartphone through AI algorithms. So these are some features of our device. So first is the smart chip. Traditionally, which we see in the market, the ultrasonic device works on piezoelectric crystal, but our chip is based on vibrating drum. So it has been more advantage as compared to the piezoelectric crystals, as it is much cheaper than the piezoelectric crystal, and it has more advanced. It can produce more advanced images as compared to this crystal, and also this. There are some studies where people have seen that there's heat sensitivity of the piezoelectric crystals. The volt it need only 10 volts, and it can be operated via smartphone using AI algorithms. The below metal helps in penetration of the sound waves, and the ultrasonic diagnostic imaging frequency is about 2 megahertz to 15 megahertz, and customization of this jewelry is possible as per the customer's need. As I said previously, removable chip is available, which makes this jewelry as wearable. And next is it's cost effective. It's cost effective because the as compared to the market available ultrasonic imaging devices, the quartz crystals are bit expensive as compared to our vibrating drum, which is made up of polymer resin. SWOT analysis. SWOT analysis is done to determine its strength, weakness, and opportunity and threats of our model. The strength of our device is that we can make medical diagnosis much quicker and easier for physician. Improve patient compliances as diagnosis can be done at home settings as per the needs. It is cost effective as it reduces medical expenses. Detection of diseases such as prenatal disease, breast cancers, thyroid disorders, Parkinson, etc. And it is time saving, hence improvise the quality of life, as it doesn't use any radiation, so it is an eco healthy system. Next is weaknesses, as we know, ultrasonic imaging doesn't produce image for bones and lungs, so we can't use this device for obtain image for bones
Someone
and
is
lungs,
speaking
and
from.
there is no pre-existing medical market model use as a commercialization when opportunities. It is beneficial for healthcare.
ज्यादा वेरिएशन आएगा तो फिर ये अलाउ कर देंगे. परफॉर्म तो करो आप.
हेलो.
बस स्टॉक रेडी होने के बाद इमीडिएटली उसमें से वॉल्यूम विड्रॉ करना है, सैंपल बनाना है. 10 मिनट का ही काम है.
Who is talking in the background? Please mute yourself. Yes, please continue.
Okay, it is beneficial for healthcare sector in poor countries or in rural areas. It is a novel tool in crisis situation. It also avail a digital platform in hence applicable for worldwide treatment of all over the globe. It also will give a low cost imaging system applicable for diverse medical application, and scale up of this medical device is possible. Threats is that market and social acceptance are still unclear, and clinical trials will be costly. Conclusion: future smart device. Yeah. Future smart jewelry having attractive design and wide applicability, enabling a medical profession to reach out all over the globe for better monitoring of healthcare. This allows for future release of jewelry's creative design in future. The purpose of this innovation is frequently represented by encouraging the creation and improvement of human-made products and motivating individuals to identify new needs and find new solutions to issues. So these are the references which we use to our research works. Thank you, and thank you for giving us this opportunity, BNCP.
Thank you, IIC5. The session is now open for Q&A.
Yeah, my first question would be, when you say that it's important or it is useful in the diagnosis of diseases, prenatal diseases. Personally, I find it a risky affair because based on the wrong practices being followed in the medical industry, you know very well, right? Detection of. So how you will tackle it? How you will tackle it? Because it is then very risky. If suppose I get this jewelry and if I start using it for the purpose which I should not be, then the basic novelty, not novelty, but the concept itself will go wrong, right? So how you will tackle it?
So basically, the image won't be read by the patient itself. He needs the help of the medical practitioner. So
right, but nowadays, sorry to interrupt you, but people are so addictive to Google, right? So a normal person may find out many avenues to read the result and self-diagnosis. Then what is the remedy for this? Self-diagnosis is worst as far as our country, anywhere in the world, because everything is available on Google. So tackling all these points will be tough for you. Explaining to even medical practitioners, because where you will sell this basically product as a medical device or what, as an instrument, or only you will target on some clinics. It will not be available in general.
Sir, our aim is to make this available in general. Like it's a jewelry, so it should be worn by each and every person with respect to the health and system.
But then I doubt, because if you are making it for general public, then sure there will be a wrong use of this jewelry, because it is very comfortable to hide from anyone. Like the sonography machines, why portable sonography machines are banned? The basic reason is that only, because of these malpractices. So taking the jewelry which you will launch in the market, it will be very easy. So you should give a thought on that part. No doubt, as an invention, it is very good idea. But when you say that you will make it for general public, then I find it's a risky proposal.
Okay.
Making it to general public. And what are the how you will validate the results? Suppose if sonography is done using this jewelry, how you will validate that your results are at par or same, whatever, with the conventional practices, which are much more now modified, or you can say the results are very much available in shortest span of time. So how you will validate your jewelry or the output against the current one?
So basically, this part is yet to be done. Like we have not validated yet, but we'll do that.
So clinical effects and everything, you will have to still you're thinking on it, right?
Yes.
But then before that, even give some thoughts on social impact. If it is generally available in the market, please. That's my sincere suggestion. Advice rather, not a suggestion. Social impact you should consider before going further, or try to make it very, I can say only a genuine medical practitioner can read the results and conclude. Okay.
Yeah, sure.
A normal person, they can wear it. It can be used in as an SOS situation where it is required, but doctors cannot reach. Then your jewelry can be of very useful, because you can go to patient, get data, immediately travel to some big cities, like from villages to big cities, get it authenticated from a genuine medical practitioner, and then advise the patient accordingly. So that I would say that would be the best approach, instead of making it available for a general public to avoid malpractice. Okay, so think on these grounds and then work on your product. Okay.
Sure. Thank you, sir.
Thank you. Thanks, madam. Over to you.
As sir rightly said, he has given very, very valid points. Rural areas where there is no diagnostic techniques reach, tools are not available. For them, it will be a major support, provided you validate it. Okay, and for the diseases which can claim lives. Okay, so this type of diagnostic tools can be helpful, so that in a timely manner they can reach the medical help. So that could be your target population, not the general public who is advanced and misuse the device. Okay, so that it's a very good suggestion. So yeah, and validation, because the output which you get from these devices and the huge devices which we have in path labs and all, or the diagnostic centers, the results should match, right? So validation is very, very important. Okay, so that's what I say. Good, congratulations. Good concern.
Just to add one point, sorry. Think on this, like if suppose jewelry is, these reports are available, then how your medical practitioner will respond in such cases. If you go with your report, device, everything to a medical practitioner, and if you ask him for a conclusion or advice, will he respond the way he will analyze or test all these whatever requirements are in his own laboratory?
Okay.
That also is a point you should consider. Okay.
Yes. Thank you.
Thank you, IIC 5. Thank you, judges. Now I request IIC 6 to be ready. IIC 6, are you there?
Yes, hello.
So, will you share the presentation or should we share the? Okay, you shared the presentation.
Yeah.
Your time starts now.
So, a warm afternoon to one and all present here. So, are you all tired of pricking your finger and putting a drop of on the blood of the test strip every time you want to measure your blood glucose level? Yes, right? That all changes right now. Viewing blood sugar with just a quick glance at your wrist is no longer a futuristic dream for people, but now a reality for many of us. This presentation today aims to review the literature on the use of wearable technology monitoring parameters that lead to the development of diabetes management. Diabetes mellitus is a metabolic disorder that affects hundreds of millions of people and causes several deaths every year. Such dramatic scenario puts some pressures on the administrations and scientific communities to seek novel solutions that may help control and deal effectively with this condition. This now reminds me of a small incident. A person named Jack was diagnosed with diabetes type 1 and was instructed to monitor glucose level 4 times in a day. Usually, he used to have his proper lunch in the office at 2 p.m. and then once he leaves the office premises at 5, he used to measure his diabetes. One fine day, such happened that despite of having a full nutritional lunch, he felt hungry at 5. He craved for a carbohydrate bar. But as soon as he measured his glucose levels, he showed that he was hyperglycemic, which is normal during the post-lunch measurement of diabetes. He panicked and failed to eat everything, which eventually led him to being hypoglycemic, and dizziness and fatigue accompanied him. So, what happens here is that the glucometer indicates your glucose levels at a particular moment and not that average range of the past few hours. It is not possible for all of us to sit and prick our finger every half an hour, right? So, to beat the age old, outdated manner, smartwatches and CGMs are the provided solution. Can we go to the next slide, please? Yeah, so over the last 2 decades, dozens of companies worldwide have sought to develop a non-invasive glucose monitor, a sensor that could measure your glucose level without puncturing the skin or having to insert a device into the body. However, majority of these devices have either failed to work or have fallen short of the FDA approval. Smartwatches cannot currently monitor blood glucose levels directly, but we can link them to an external CGM. A CGM is a continuous monitoring device which connects to the smartwatch. Watches such as Samsung Galaxy, Apple, etc. provide the function of connecting with the third-party CGM, which eventually lead to let the blood sugar data on the wrist. A glucose sensor is a CGM device and it is often positioned under the belly or under the underarm. It gauges with the glucose in the interstitial fluid, and the receptors there send the signals to the blood glucose reading device, and from there it is connected to an output such as watches. Can we go to the next slide, please? Yeah, so apart from CGM, other non-invasive mechanisms that can work are a monitor that shines a beam of light through your skin, which is then reflected and scattered by the glucose in the blood. The sensor then detects the signals from the light and is reflected black. A glucose exhibits a unique reflection of the pattern that differs from the other components of the blood, such as potassium, calcium, etc. So the difference in the signals is how we can measure the glucose level of the blood. Of course, this is very much easier said than done, but they hold a variety of advantages, such as sensors can avoid the simple basic pain of the needle. It can eliminate false readings due to contamination by the substances on the surface of our hands. And we can also think about the environment and limit the waste from disposable testing supplies such as strips, landslits, etc. Additionally, many non-invasive monitors have no disposable components, which could wipe out recurring costs. Can we go to the next slide, please? Yeah, so the given picture shows the representation of the generations of diabetes monitoring over the years. Now, the non-invasive techniques are still looking for the development. The average cost of the product that we are proposing to the market could range between 30 to 35,000, and the target audience will be the patients having type 1 diabetes or who have hypoglycemia unawareness, people who have very frequent fluctuations in the glucose level, or the people who have intensive insulin therapy. Can we go to the next slide, please? Yeah, so in conclusion, smartwatches are the future of non invasive diabetes monitoring. It records all the spikes we experience through diet and alarms us at even the slightest change. As life is uncertain, the best we can do is to look out for ourselves and our loved ones. Therefore, I advise you all to go ahead, take a charge, and look at health at your hand. Thank you.
Thank you, ILC6. Now the session is available for Q&A.
Yeah, my first question would be, will iWatch will be your competitor?
iWatches currently do not measure the blood glucose level directly from the.
They have plans. They have plans.
Yeah, they have future plans. They are still working on it.
So how you would rate yourself against iWatch?
Because iWatches are very expensive compared to, like, not all the normal population can afford iWatches, and iWatches can only connect to the products which are associated with Apple. And in current situation also, iWatches, not all iWatches can connect to the CGMs.
Right. So taking inspiration from iWatch, other competitors, or even you can say BOT, which is total. I believe it's Indian company, right? If
Yes,
they
the
take
one.
this challenge, yeah.
One basic advantage that we have over others is that it will be very cost effective as compared to the others, and we will also install an alarm which alarms you whenever the blood glucose level fluctuates. That is not what has been happening till now, and yeah.
Okay, but it will be battery operated again, right?
Yeah, it will be battery operated, and the basic battery life will be around 38 to 40 hours.
Then have you validated like start of the battery, means first time installation, and after 48 hours, how it will give you indication? You should validate that with actual reading. Okay, but because in general, a blood pressure monitor is available which is battery operated, and if the battery goes weak, there is definitely a drastic, or you can say not that drastic, but there is a change in blood pressure reading I'm talking about. So you get a false alarm. So how you will deal with, because blood pressure, suppose I would say okay chalo, if it is 120 80 ki jagah agar 110 and 70 aaya, then patient will not be that worried. But in case of your smartwatch, if the sugar levels are, because of battery weakness in the battery, if sugar levels are indicating low results, patient is happy, he will go in Navratri, he will enjoy the sweets and all, and suddenly in the night something goes wrong.
Yeah, so
How you will deal
we'll
with it?
advise them to charge, as like we'll have a battery life indicator also. Like once the battery goes below 20%, they'll indicate when it's about to die.
Suppose I'm traveling to US.
Okay.
24 hours journey total. Flight is almost 14 hours. And if the battery is already 8 hours old, okay, how I should deal with it?
So we'll have a portable charger also according in the packaging. We'll give them a portable charger so they can charge as and when the battery dies.
And how you will validate the readings all 40 hours?
For the validation of the readings, as we are as prescribed, the person has to prick 4 times a day, right? So it is advised that along with the watches, instead of 4 times, just check one time in 24 hours, so that if the readings match, you know that it's going in the right direction.
Have you not given any thought on solar charging?
Yes, sir, but it is not possible on the places where we don't have solar energy, or where there are harsh climatic conditions, or during monsoon. So it is not possible.
Why not as an option, alternate?
Yeah, we
So
can
that
consider it.
if you definitely the patient will keep charger, but at the same time, if he's traveling, definitely because of solar energy, the battery will be in condition. Why don't you think on that lines?
Yeah, just as I mentioned that during monsoon or during
If
periods
you
like
have observed by any chance calculators nowadays, there is a solar patch. Have you observed on Casio on the left side of the calculator?
Yeah. So yeah, we could think along those lines as well.
Out of box thinking.
Yeah.
Think. Okay, ma'am, please. I'm done.
No questions from my side. You can go ahead.
Thank you, ma'am.
Thank you, IIC 6. Thank you, judges. Now I request IIC 7 to be ready. IIC 7, are you there?
Yes, sir. We are there.
Yes, sir.
So, will you share the presentation or should we share the presentation?
Sir,
Yes.
I'll share it.
Okay, no worries.
So is it visible?
Yes, it is visible. Time starts now.
Okay. First of all, a very good afternoon to everyone present today. Today we are going to elaborate on our idea of Aqua Rhizo 9. It is a bio-inspired heavy metal chelation assembly that will help in stripping off the industrial wastewater from the lead and arsenic impurities, and will also help in promoting the bio-entrepreneurship, which will be one of the contributions in the Atmanirbhar Bharat project. We are abstract number IIC07. And if let's start with the introduction. So we are going to challenge two problem statements. The first one is industrial wastewater that often contains heavy metals are also containing harsh chemicals like acid, soaps, and solvents, at times different kind of detergents and buffer solutions. So we are going to think upon how we could bring a sustainable solution to address this issue so that the industrial wastewater could be treated effectively. The second problem statement that we are going to address is we are using two plants. That is, here we have named them as X1 and Y1 because of patenting rights. We are not allowed to disclose their names, but these are their pictures. So these both plants cause eutrophication, and we are going to try to repurpose these both plants, and we are going to help in solving the problem statement 1, that is industrial wastewater. We are going to do it with the help of phytoremediation. By definition, phytoremediation is nothing but use of green plants or microbes that helps in concentrating or breakdowning of pollutants so that the pollution level of water, air, or soil could be reduced sustainably. Now, our hypothesis here is we are going to use phytoconstituents present in X1 and Y1 plants, roots or the non-aerial parts, and we are going to chelate it with the toxic heavy metal impurities that are present in the industrial wastewater or different kind of catalysts which are involved during the process of manufacturing, and a few of also other agricultural impurities like neonicotinoids and dibutyl phthalate. Right now, due to extensive use of fertilizers, even the soil pollution is increasing daily, and agricultural wastewater or the water which is leaching out from agricultural lands are seeping into the soil, deep into the groundwater levels, and eventually meeting the river bedside. So we are going to channelize all these ideas and we are going to put it into our hypothesis. And after doing a chelation complex, we are going to do a water insoluble complex which will precipitate down eventually in our filtration device, and then the supernatant.
Excuse me, sorry to interrupt, but can you share the slideshow mode?
Ritika, the slides are not moving. That's here.
Yeah.
Okay, just a second.
We couldn't see the PPT right now.
Is it visible
just
right
give
now?
sir.
Yes, it's visible right now.
Oh, it's visible now. Okay.
Put
I'm
it on
so
the
sorry.
slideshow.
It's on the slideshow.
Full screen. We want.
Yeah, it's on the full screen. Amay, could you share it for me?
Okay, just a sec. You stop sharing. I will do it. Wait. Is it visible?
Yep.
Yes, it's visible.
Okay, just a second. Slideshow from beginning. Okay, so Ritika, you are on which slide?
Slide number 2. Yeah, thank you. Yes. Rightly what I was talking about, like we are going to use the principle of chelation because a few of the properties or the phytoconstituents of X1 and Y1 plants tend to form chelation or chelating compounds with the heavy metal impurities. And the complex that would be formed would be a water insoluble complex, so it would not again leach out into the water body system. And then like the next slide. Then these are the objectives of our project. We are going to laboratorically prove that phytoremission abilities of both plants, and we are going to quantify the rate of absorption with respect to time and the biomass for these two plants. Now, we are also planning to extract, isolate, and identify and structurally elucidate the responsible phytoconstituents. Right now, there are not much studies carried out on these two plants because these are considered as an invasive species. They cause hindrance in the balance of aquatic ecosystem, as well as to some extent it contributes to pollution with respect to aquatic life. So we are going to go a step ahead, and we're trying to repurpose by isolating and extracting different phytoconstituents that could be capable of forming a chelation compound with heavy metals. Now we are trying to also find encouraging like repurposing ideas for X1 and Y1 plants so that more people could be like aware of how they could be repurposed. Like recently, these two plants are also being used for mushroom cultivation. So, like other than this, they also have some application in vitamin C serum making processes, and so we are also trying to enlighten it to a step ahead in water purification system. Now, the last thing is we are trying to prepare an industrial device that will help us in reducing the lead and arsenic impurities which are leached out from industrial effluents into the wastewater. Next slide. Now, how our project is relevant to the healthcare system. As it is rightly said that health is not only involved with respect to just how the food you intake, it is also very much similar to what kind of liquid diet you pursue. So the majority of the project that we are going to target for the audience is going to be the rural population, because they really have lack of access, they have lack of opportunities towards better healthcare systems, and also they are not financially that stable. So if we use our project to clean the water body systems or resources which are nearby the rural population, the water supply that they would get eventually would be much more potable, and their lifestyle and healthcare would improve substantially. There are different kind of diseases which are like related to PB, that is lead and arsenic. So we are going to try to address those. Now, these are the experiments, which was the initial part for our hypothesis. We tried this assembly of 4 chambers here, that is pre-filter, central chamber, and sediment filter. And we incorporated our central chamber with some novelty idea of those X and Y plant phytoconstituents. And the drawback of this project was that even though the chelation was happening, due to the entire usage of root powder, there was clogging going on between the central and sediment chamber. And even the sediment filter was to be replenished very quickly. So we tried to change the entire approach. We visited a scientist from NCL, Dr. Seema Ghate. She's a phytoremediation scientist, and she like gave us few inputs on how we could remodelize our idea and we could move ahead. Further, Amay would continue. Thank you so much.
So after the failure of this initial approach of ours, which was for targeting drinking water, we decided to target industrial effluent enriched water that too just before it is about to be released in lakes and streams. So our first experiment was taking limited unit quantities of that water, introducing the plants in them, and then introducing stock solutions containing fixed concentrations of both the target heavy metals, which are lead and arsenic. So for arsenic, we calculated the average concentration in industrial effluents, which is 35 milligrams per deciliter, and for lead it is 42 milligrams per deciliter. So to we started testing with lead, and with that we introduced those stock solutions, and then after the unit amounts of time, like 2 hours, 4 hours, 24 hours, we withdrew small quantities of water samples from those water buckets in which the plants and the heavy metal solutions were introduced, and we checked them on UV. So now a bit of context on UV, lead acetate shows absorption maximum at 210 to 215 nanometers. So with that, we use the calibration curve method to quantify what is the absorbance at initial T is equal to 0 hours, and then how absorption goes on reducing progressively with time. And we correlated that with the concentration to quantify how much exactly the plants were absorbing. Our second experiment was focused around which parts of the hello. Sorry.
2 minutes are remaining.
Our next part was focused around quantifying which part, either whether the aerial parts absorb more or the non-aerial parts absorb more. So we subjected both of these parts to combination and centrifugation, and that was at the upper part of both the centrifuging bottles that was analyzed, and we also from that we found out that the roots were doing the major weightlifting when it comes to absorption, and the leaves only contributed later on when it came to storage of the impurity later on, as in by the parenchyma tissues. And these are the future experiments that we are planning to carry out. So arsenic stock solution preparation, because getting a soluble salt of arsenic is unfortunately posing itself as a problem for us. So we would want to reproduce the results that we have produced with lead with arsenic as well. The next part would be to carry out, as in once again carry out plant sample analysis this via other labs. Like we have contacted one lab in Pune called Mark Labs. So we are going to send our water samples as well as plant samples to them for analysis to reproduce this with their highly accurate devices. Next, we are going to carry out complexation studies and molecular docking studies to find out which phytoconstituents have the highest binding affinity.
Your time is up.
Okay, I'll try to summarize it in a minute. So we want to find out which phytoconstituents selectively bind with both the impurities and chelate them effectively using these two approaches. And then we want to create a device which is very reminiscent to rotatory drum filter, such that if industrial water is put in it and it is rotated for a limited period of time and then sent out, the output water would have no heavy metal in it, like arsenic or lead. So these are our conclusions, and these are our references, and that would be it. Thank you so much. Just a second, let me.
Thank you, IIT7. The session is now available for Q&A.
Wonderful presentation both. I was actually waiting how you are concluding it. Apart from this judgment and all, if you both get a time, I would like to get a proper more presentation in our R&D center whenever you get time. Okay, I would love to get some more information on the topic which you are working on right now.
Yes, sir.
Okay.
Definitely, sir. Definitely. It's a pleasure.
Yeah, it's a wonderful topic, wonderful presentation. I have no comments as on date because you got some place to represent, but probably a wonderful presentation, perfect presentation, I would say, because these are the analysis part, and we have to consider more critically if you are talking to industries.
Yes,
effluent treatment is a hot topic, environmental monitoring and all. So I would be happy to interact with you further on this, just for our knowledge. Okay.
Yes.
What you are working on, so that if possible,
Yes.
if we can implement this in the R&D center, I would be really happy.
Yes, sir. Thank you so much.
Definitely.
Thank you so much.
Thank you so much. Thank you.
Yeah, good presentation. Just wanted to know this lead acetate, no? The degrees which you have got, it's 10-fold decrease, right? So how many times you have repeated this experiment?
So we repeated it with other molecular candidates that were already out there, like lead acetate, lead sulfate, and we are currently trying to recreate it with lead oxide as well, because that is one of the most common
You're
lead.
getting similar type of decline in the levels.
So in sulfate, we are getting slightly lower decline compared to lead acetate, and for lead oxide, the results are yet to be drawn. Like we are just started carrying out right now, and for arsenic, the difficult. Sorry.
Yeah, it's basically lead ion, no, which is there, and you're adding acetic acid. So you're making lead acetate for the analysis purpose. For the
Yeah,
analysis.
yeah, we're directly using lead acetate powder, and during stock solution preparation in 100 ml, we have to add 1 to 2 ml of acetic acid for a better dissolution as well.
Yeah, but the sample, the wastewater, which after treatment with the plant, in that you are adding acetic acid or no? How are you going? How are you taking the absorbance?
Taking absorbance, as in
Decrease in lead acetate. How are you going to estimate?
Yeah, so after we withdraw small quantities of water samples, we are taking them via UV. So we are doing serial dilution by factors of 1 by 1000, as in if we would take 1 ml of the withdrawn water sample, we obviously stir it before we withdraw the water sample, and then we take that in a 100 ml volumetric flask, make the volume to till 100 ml, and then after shaking, we also once again take 1 ml of that and put it
Ajay, you simply dilute it and then you take the absorbance at that wavelength, right? Because the wavelength that you're selecting, no, it's the wavelength where a lot of gets absorbed. Okay. So you just see the interference at that wavelength.
Yeah, now I can hear you.
Interference at that wavelength because the 210 nanometers in UV. Okay, some maybe other technique like atomic absorption spectroscopy or something, which will give you a precise, accurate result to conclude on. That is one solution because lead acetate having the weak chromophore to analyze at UV becomes to conclude because that is your result, right? And one more thing, because you're removing sample from the tub, how much sample you're removing, you got to replenish it in order to see, like you do in vitro drug release, right? Something similar to that. So replenishment, the sample has to be done because it's all quantity. You're playing in terms of quantity, right? So your analysis has to be accurate if you want to claim because you're claiming reduction here. Okay. So you got to take care of all the error contributing factors in the analysis. So that's it from my side. Thank you.
Yes, ma'am. We'll keep it in our mind next time. Thank you so much.
We'd love to connect with you on.
Thank you.
Thank you, IIC
Thank you so
7.
much for the
Thank you,
opportunity.
judges. Thank you. I request IIC 8 to be ready. IIC 8, are you there?
Yes.
Okay. So do you want us to share the presentation or will you share the presentation?
I'm sharing.
Okay.
There is echo here. Two devices are on.
I'll check. Hello?
Yeah. Share your screen.
Is it okay now?
Yeah, it's fine now. With the voice, share your screen.
IIC 8, are you facing any problems or should we share the screen if you want?
Yes, dear, we can share the screen for you. You can just present then. Please respond. Visible? Not yet.
No,
I
it's
think there
not.
is some network issue. We can share the screen for you. In the respect of time, you can present then. Kunj, you can share the screen.
Okay, ma. We are sharing in a few minutes. Is it visible to you? I see it.
I see it. Please respond.
I see it. Are you there?
It is visible to me.
Okay.
Okay, please go ahead.
Your time starts now.
Yes, okay. Good afternoon, everyone. My topic for Innovative Idea Challenge is mouth dissolving films of caffeine for driver fatigue. Next slide, please. So the problem I'm focusing now. Hello? Hello?
Go ahead,
Yes,
please.
We
We can hear
Go
you.
ahead. We are.
The
Yeah, we are.
Okay. The problem I'm facing that I'm discussing today is driver fatigue. So tiredness is experienced by driver due to lack of rest, and statistics say that almost 2 to 3% of road fatalities and 13% of road accidents are caused due to driver fatigue. So what happens is there is decreased alertness and increased reaction time, because of which the driver can lose control and result in road mishap. Next slide. So there are various causes of driver fatigue. Few of them are listed. That is insufficient and poor quality of sleep, because there is long working hours and work shifts. It can also be due to disruption of their daily routine or environmental stresses. Besides these, medical conditions are also responsible for causing fatigue. Next. So, the solution that I thought of is caffeine films for drowsy drivers. So, caffeine being a CNS stimulant is a BCS class 1 molecule which is good solubility and permeability. So, it can be formulated in mouth dissolving films, which can be used as a instant stimulant. What are the advantages of such a formulation? They are easy to carry, and there is no need to take a break during the driving for sipping a cup of coffee. Besides that, it is also instant action because the film will be rapidly dissolving. Next. This is the proposed methodology wherein the key ingredients of the formula are listed. I'm planning to load 50 mg of caffeine per film because that is the stimulant, and the polymer that will be used as a film former is toluene. Other polymers can also be used, and the solvent for all this will be water. So the proposed methodology goes as solvent casting technique, wherein the blend of the active, the polymer, and other excipients are made into the vehicle, and this blend is casted in molds or trays, and after that drying can be done, and then the films are cut into the desired shape. Next. So these will be the evaluation parameters for caffeine-loaded films: the thickness, tensile strength, content uniformity, endurance, in vitro drug release, and stability studies. The films are to be kept upon the tongue, and after which there will be release of caffeine. These are the next slide. Okay. So the approximate cost which I came up with was 5 rupees per film. And besides that, the advantage of this is there are no volatile organic solvents used. The polymer used is biodegradable and GRAS approved. And the method of preparation is also simple. And there are no toxic ingredients and no volatile chemicals, so there is no problem of flammability, and hence it is easy to scale up an industry friendly formulation. Next. So besides driver fatigue, this formulation can also be a favorable option for people working in the disaster management or emergency workers and truck drivers. It can prevent fatigue-related road mishaps, but this is a solution for short or acute. This is a solution for acute duration. It cannot be used for chronic duration because caffeine may lead to sleeplessness uh, and other problems. And it has applicability to other fatigue-related situations as well. So this concludes my presentation. The next slide is about the references. Next slide. These are my references, and thank you.
Thank you, IC8. The session is now open for a Q&A session.
Yeah, what is your target customer? Who is your, sorry, who is your target customer basically?
Yes, sir. So the target customer as of now are drivers who are involved in continuously driving their vehicles. So professional drivers, truck drivers, etc.
On lighter notes, I'm not discouraging you, but all these truck drivers and all, they are addicted to either a cigarette or weed, which is much more stronger than caffeine oral film. What do you think on that? So what about their education, use of this film? And if I'm considering a population like us, educated population, then Red Bull is available in the market, which is around 35% of caffeine. So what do you think on these two grounds? And my third question is most important. You have used pullulan as a gelling agent, right, in the film? And you said the
as
cost
a film former.
Yeah, as a film former. And you said the cost of film per patch is around 5 rupees. I doubt whether if you can manage in 5 rupees, because suppose 25 or 100 gram, 25 to 50 grams of pullulan is costing around 11,000 rupees. It is very costly gelling agent. So you have to rethink on your costing part. If you are not aware of actual cost of pullulan, if you have received any gift sample for your work, and accordingly you have thought of 5 rupees as a pricing, then think on these grounds, because pullulan is very costly. In the industry also, it is not used so well as a gelling agent till date because of costing. In the generic industry, we are very much concerned about cost, and we haven't used or tried this despite having good properties. I accept that it's a very good gelling agent which you have selected, but considering the cost which you are thinking, this may not be an appropriate cost. So rethink on that. So all these points.
Okay, if I can answer your previous questions. Firstly, the smoking and the thing that you said, so they shouldn't be using it, and basically they are using it and they are using their hands for smoking or anything like that. So they are distracted basically. So using these films, they are just supposed to keep the film on their tongue and it will dissolve and give them the stimulant effect that they need for driving and increasing their alertness. Then about the Red Bull also they have to stop and drink the can and carry the cans wherever they go. So that is bulky. Whereas films can be slipped into the pocket and it is less bulky and easy to use when you want to go and travel. Then about pullulan, I found a vendor that gave pullulan at a lower cost, and hence I estimated the price, but I'll surely check, sir. Besides pullulan, other polymers like HPMC, HEC can also be used. Besides, Udrajet can also be used.
You have not considered about the manufacturing facility. Eh? Right now, there are very limited facilities in India, and it needs a volume business, not a value business. If there is a good volume, then only you will get the value for your product. That's the logic of this business. Oral films.
Okay.
That's why you can see there are no many players in the oral film industry. So if at all you are planning, you can sell your concept to Zim Laboratories located in Nagpur. Just note down. They have biggest manufacturing facility, and you can get some good ideas from them. If at all they have already caffeine patch in the product list, then I'm sorry you will be discouraged with your project. But why not to approach and check with them? Because machinery and all this cost is very high for oral films. Keep this in mind.
Okay. Sir, I'll surely
Yes,
check.
ma'am, please. Yeah.
Yeah, I just had two questions here based on Pululan only. Pululan, these drivers which you are targeting, if they are suffering with diabetes, high glucose levels. So Pululan being a carbohydrate, just see what is the impact of that. Mostly, I think it will be slowly digesting carbohydrate. So that consideration has to be done. And because recently I was researching about this Pululan, it has some GI problems like some flatulence or something like that as a side effect. So if it is taken on the empty stomach, so and repeatedly, frequently, maybe 4 films per day, so that type of effect could be there. So even that has to be looked upon. Basically, because Pululan you used as a polymer. Okay, so if there is any GI side effects, because patient is using it for patient or the consumer is using it for being awake, with that there should not be any GI disturbance, no? So
right, ma'am. So, I have the safety data of Pululan. So it says that almost 10% Pululan is equivalent to 7.9 gram per day. So it is safe.
It's not about safety and toxicity. It's about side effect. Everything on this earth has some or the other effect, no? So it's the side effect. I'm not talking about safety or toxicity. Okay, so that's from the literature I'm saying. You can just take that into consideration when you're formulating your product. And additionally, of course, as the sir said, cost is important. Your product should be affordable to the consumer, right? So if you can replace it with a polymer which is, bringing it to the cost that you desire, then it will be great.
Okay, because I did find a company which gave Pululan at a lower cost and hence estimated that price. But I'll surely take yours and sir's suggestion.
It should be orally edible also, no? The low cost grade. Even that we want to check. Yeah. So that's from my side. Thank you very much. Kunj, you can take it over.
Thank you, ma'am. Thank you, sir.
Thank you so much, IC8. Thank you, judges. I would request our judges to provide a brief message regarding the session.
Yes, sir, please.
Yeah, it's really very well organized session and interesting as well, because no doubts as an industry person, I have many doubts which are say like scalability of the project or the thought process, whether it will work in the industry or not. Accordingly, I was thinking for all these projects, but no doubt students have given a good, they have spent a good time and given some valuable presentations. Really, I'm happy. Encourage them. That's why this last person, ISC 7, I think. 7, right? Yeah. I would like to get some more details from them.
Yes, sir. Yes.
It's really wonderful. They couldn't get much time because of the limitations, but it's really a nice work which they have done. If they have approached to any consultant or expert, it's really good job what they have done as a student.
Thank you,
as
sir.
An
Thank you, sir.
Thank you, sir.
So I would like to interact with you more. No doubt all projects are very good. There will be plus and minus in our judgment based on what we think. It's not, we are not giving any ranks or priority or anything to the projects. It's only the thought process of students, how they have presented, will it be workable in the industry, scalability, all these factors I have considered. So don't get discouraged. All 8 presentations are wonderful, but the style of presentation, the thought process, everything matters a lot when we are judging from other side. Okay, so all the best to everyone. Thank you so much.
Thank you, sir. So here a lot of presentations. We had almost 8 presentations here, and each one was a new thought process and a lot of thinking literature students have done, and we have also got a lot of insights from that. So keep it up, students. It's nurturing to your young minds. Get the solutions for the problems, and do good in research. That's what the intention of this event was, to motivate your brains to think in a unique direction, and we at BNCP have successfully done that. So thank you very much, students, and keep it up. All the best.
Thank you, ma'am. Thank you, sir. Mundira ma'am, do you like to speak something?
Yeah, sure, Kunj. Thank you. The presentations, this all what both the judges said about it or commented on it is very right. The first thing I would like to say is participation is winning. All of you participated into this competition. Itself shows your winning side. So you are the winner because you participated and you competed. It does not matter whether you come into the rank or not. The mere presenting your details and innovation was very important. So thank you for submitting all your innovation ideas and presenting, and some of them were quite thought-provoking. And as a student, Dr. Pankaj rightly mentioned that as a student, they have really done a good job. And always remember that whenever there is an innovation, we always need to look at the business potential also. So thank you, judges, for giving your valuable feedback. And I'm sure the students, those who have participated, as well as students, those who are listening to the entire day presentation for innovation in healthcare, I'm sure they also have got good insight and motivated and encouraged to do something different and more scientific inputs in whatever they study and be more inquisitive. So thank you, Dr. Pankaj, and thank you, Dr. Vasanthi, for your valuable time and your wonderful comments. We really appreciate your effort for evaluating our students.
Thank you. Thank you so much.
Thank you. Over to you, Kunj.
Thank you, ma'am. Thank you, sir. The feedback form is posted on the chat box. Everyone is requested to fill the form.
While they fill up the form, Kunj, let me take this opportunity to thank my entire group of students right now sitting in the technical room. I visited that room multiple times today, and I saw such a wonderfully students are managing the entire show. So thank you, students group. Don't leave college. We are going to have something for you. We'll have tea and something refreshment for all of you. The hard work which you have done. Thank you, Lokesh sir, for coordinating the entire work. We really appreciate you guiding students. And this is the wonderful training what students get while they are in the college. The team building as well as the leadership quality and the resource management, the way time management, resource management, and the entire conduct of the event, the way they have handled technically, though they are a pharma student, they have shown their IT side also. So thank you very much, all my volunteers. We would definitely be happy to see all of you. So Kunj probably can take a group picture from the front and share the screen of his mobile or anybody probably can share, or you have a smart board in front of you. I know you have kept the smart board also on the screen, which has a camera. You can show all of you sitting into the technical room. So thank you. I really appreciate your efforts which you have put in for making this entire event successful. Without you, it would not have been. So thank you very much.
Thank you so much, ma'am. We will click the pictures during valedictory session. Okay, so I would now like the judges to receive a small token of appreciation from us.
Your thought of calling us is an appreciation.
Thank you, Doc. Thank you, Dr. Pankaj Manpre, sir. Thank you, Dr. Vasanti Suvarna, ma'am.
Thank you. Thank you, sir, for your valuable presence here.
My pleasure, always.
Great insights we have got, and it was very nice to interact with you, sir. To hear you, rather.
Thank you so much.
Thank you to all the judges for being part of this event and for providing the constructive criticism, which not just the participant but we have all learned from. And of course, thank you to all the participants without whom the event would not have been possible. It was such an inspiring and dynamic event because of the research, the innovations presented by everyone. As it is said, anything that a scientist discovers or invents is based on the knowledge of the previous discoveries and inventions. And this, the third Student Research Congress event, has been all about that. From the third day Student Research Congress event, the researchers in us has been inspired, motivated, and encouraged, and to provide such great work that will help the community to grow. Because the best way to predict future is to create it. We will be taking a break now and meeting at 4:30 for valedictory session. Thank you, everyone.
We'll be starting the session in the next 5 minutes. Good afternoon, everyone. I, Santha Zada, would like to welcome back everyone to the second half of day 3 of third Student Research Congress. As it is rightly said, the beginning is a word and the end is a silence. And in between are all the stories. And here we are at the epilogue of such an extraordinary and wonderful event. Now, I would like to call upon our Honorable Principal Ma'am to share wisdom and uplift us with your valuable words.
Thank you very much, Vaishnavi. And welcome everyone back on the last event, valedictory and much awaited session of declaring the results. I always said into the previous session and here also that winning is participating. You are a winner by participating it. So whoever is a winner, the one who did not win, do not get disheartened. You are a winner just by participation, by making an effort, competing at a national level. As in my inaugural, I very clearly mentioned that we had 781 registration. I also mentioned that we had abstracts from 39 cities. So imagine that you have competed across India and 11 states. So 18 states where the participation registration was from 18 states and abstract submission were from 11 states. You can imagine what how much effort and what you have done. You are a winner. So I know everyone is looking forward to the result, but let me take this opportunity to thank all the participants and the presenters and my entire team of students, volunteer who have managed the entire show. I really appreciate their efforts. All the judges who have taken time out of their busy schedule and critically evaluated the posters as well as oral entries. So we had a lot of posters of PG which we had put up on the website, that is bncpsrc.com website, the dedicated website for Student Research Congress. And our judges had evaluated all those posters and then shortlisted posters where further presenting their work. They presented yesterday their work. So I really appreciate their time and efforts and sincere efforts which they have made for giving the sincere efforts. So thank you to the entire team of students, the participants, judges, the registrant and the presenter, along with my entire team of BNCP faculty. We really appreciate your efforts and dedication to make this event a success. This event would not have been so seamless without your wonderful coordination with each other and your dedicated time and your inputs and a wonderful speech all of you have prepared. I'm so much proud of my students and proud of the entire faculty and team of BNCP. So all the best to all the presenters and thank you to all the registrants. So over to you, Vaishnavi. Thank you, everybody.
Yes, thank you so much, ma'am, for your kind words. And I'm sure we all must have gained a plethora of insights from you. So, saying that productivity is a result of commitment to excellence, intelligence, planning, and focused effort. To give such few highlights of our third Student Research Congress, I would now like to call upon Vaishnavi Golatkar to present the summary report.
Good morning, everyone. I'm Vaishnavi Gulatkar. So, before moving on to the summary report, I would like to share a glimpse of the third SRC. Once again, I'd like to thank all the judges and the participants. Now, moving on to the summary report of the third Student Research Congress. So, after two successful SRCs, a third Student Research Congress was organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by University of Mumbai, industry partner, ACG Worldwide, and SciTech Center. The theme for the third Student Research Congress was Innovations in Healthcare. It was a 3-day event with Day 1, PhD and PG oral presentations, Day 2, poster presentations, and Day 3, UG oral presentations and Innovative Idea Challenge. In this event, we had over 780 registrations from 18 states and about 49 cities across India, and around 60 College of Pharmacy, Science and Technology disciplines participating. A total of 239 abstracts were received, which were scrutinized based on quality and innovations in health and science, and only about 45% of the abstracts got selected. The inauguration of the third SRC was conducted offline on 28 September 2022 at the Virtual Seminar Hall, BNCP, Mumbai. The event started with a warm welcome of the dignitaries on the dais, followed by which there was National Anthem and Saraswati Vandana, as they marked paying respect to both our country and the goddess of knowledge. The chief guest, Dr. Anuradha Majumdar Ma'am, guest of honor, Dr. Jnanadeva Bhatt Sir, and two special guests from ACG, Dr. Parizat Elshidana Ma'am and Mr. Ketan Pawar, and our SRC coordinator, Dr. Lokesh Bhatt Sir, were welcomed by our very own principal ma'am, Dr. Munira Movin. Our principal was welcomed by Dr. Lokesh Bhatt Sir, after which she presented the welcome address. Ma'am's address was so energizing that it filled everyone with new zeal and motivation. We also had our guest of honor, Dr. Jnanadeva Bhatt Sir, address our audience. He threw light on what innovations are about and how the bridge between the academic and industry can be bridged. Dr. Anuradha Majumdar Ma'am, the chief guest for the day, gave a marvelous speech and awed everyone by explaining how important Mother Nature is, the value of good student-teacher relationship, and the impact research has on the lives of the people. It was really a bliss listening to her insightful talk. We also had small talks from the special guest, Dr. Parisat Elchidana Ma'am and Mr. Ketan Parmar Sir. Later, all the dignitaries were presented with the memento by Dr. Munira Momin Ma'am, followed by which the whole event taken on to complete online mode. The 3-day event of Third Student Research Congress conducted from 28 September 2022 to 30th September 2022 was filled with a profusion and a range of research works and innovative ideas coming from across the country. The event was streamed online on MS Team and YouTube. On day 1, the inauguration ceremony was followed by 2 sessions for the PhD oral presentation competition and PG oral presentation competition sessions. The judges for both the sessions assessed the presentations carefully and provided their insightful inputs and valuable feedbacks to all the participants. On day 2, we had poster presentation competition divided into 2 categories, PhD, PG and UG category. All the participants displayed incredible research works on some of the significant topics. The judges for both the sessions submitted on the best abstracts, selected the best abstracts from overwhelming number of abstracts submitted on the bncp.src website. The research work of selected abstracts was presented on the online platform. The participants presented some remarkable research works. The judges for the session provided their valuable suggestions and their inputs to all the participants. On day 3, we had oral presentation competition by the UG category in the first half and the Innovative Idea Challenge in the second half. The participants presented some excellent research works and exceptional innovative ideas. The judges for both the sessions assessed the presentation and provided constructive criticism by being active part of the event. With this, I would like to conclude the seminar report for the third Student Research Congress. So now, moving on to the valedictory section. I'm sure everyone's having an adrenaline rush as it is the most awaited and breathtaking moment of the event. Now, I would like to call our very own principal, Dr. Munira Momin ma'am, to announce the winners of the PhD oral presentation competition. Ma'am, just a minute, we're sharing the presentation. Ma'am, you can announce the winners for the first category, PhD oral presentation competition.
All right, so congratulations to all the winners. I'll start from last. So third prize for PhD oral goes to Shrusti Lukerwale Shubham Banerjee from Naipur, Guwahati, Assam. The second prize goes to Vivek Yadav Sanyog Jain from Naipur, Mohali, Punjab. And the first prize goes to Reena Sharma, Varun Kushwa, Samir Katiyar, Chandar Prakash Dora, and Sanjog Jain, Naipur, Mohali, Punjab. Congratulations to the entire team of all three prizes, and kudos to your mentor and your hard work. You really presented so well, and it actually created a lot of discussion, and the scientific inputs came from all the judges and all the three groups. They responded very well. Congratulations to all the winners.
Thank you, ma'am. Congratulations to all the winners. Now, I will request ma'am, Dr. Munira Momin ma'am, to please present the winners for the PG oral presentation competition.
So here, let me tell you before the name appears, if you can probably stop this and do not disclose this name. So here for the PG oral, quite a tough decision the team of judges had to take. There was a lot of deliberation between the judges, lot of discussion between the judges, and ultimately judges came up with the decision that they will not be PG oral. So the only winner from PG oral is first prize, and the only prize goes to Rajnish Prajapati and Sayani Bhattacharya from Krupanidhi College of Pharmacy, Bangalore, Karnataka. Congratulations to the team, Krupanidhi.
Thank you so much, ma'am. Congratulations to the winners. Now, moving on to day 2, I would like to ask Sujata Savarkar ma'am to announce the winners for PhD PG poster presentation competition.
Okay, it's a really proud and overwhelming moment for all of us. Congratulations to the students, congratulations to all the participants. And as rightly Principal Madam said that you got selected for the presentation, you yourself are winners. But since considering the format, one has to be first and second. But according to us, all of you who have participated, presented your novel ideas, we really enjoyed all 3 days. It was a very good learning, a lot of knowledge sharing session for us. So without much ado, let me announce the winners for PG PhD poster presentation, beginning with third position. So we have at third Neha Anwar Desai, mentored by Professor Munira Mumin of SVKM's Dr. Banuben Nanavati College of Pharmacy. Congratulations, Neha and Madam. Now, for the second position, we have Priya Shejul, Parnika Sos, Neerav Bhatia, and mentored by Dr. Gaurav Doshi of SVKM's Dr. Bhanuben Nanavati College of Pharmacy. Congratulations, team. Congratulations, mentors. And at the first position, we have Zamir Khan of HR Patel Institute of Pharmaceutical Education and Research, Shirpur District, Dhule, Maharashtra. Congratulations, all the winners.
Thank you, ma'am. Congratulations to the winners. Now, I request Dr. Sujata Savarkar, ma'am, to announce the winners of UG poster presentation competition.
Okay, now for UG poster, I'm sure again the judges must have found a tough situation. They must have been in dilemma whom to announce. But firstly, let me congratulate all the students who have participated at UG level, because that has inculcated, I'm sure your participation really must have inculcated, and churned a lot of thoughts that how can I take this research ahead for PG and subsequently PhD. And I'm sure that must have been a very good learning experience for you all. So over to third prize. For third prize, we have Palak Nagori and Ketki Karalkar from SVKM's Dr. Bhanuben Nanavati College of Pharmacy as the winners. So congratulations, Palak and Ketki, for your achievement. At second prize, we have Vinota and Niveta V and Kokila from Vellalar College of Pharmacy, Erode, Tamil Nadu. So congratulations, team. And at the first position, we have Pramodita Shende, Purushottam Gangane of Dada Saheb Balpande College of Pharmacy, Nagpur, Maharashtra. So congratulations to all the winners and congratulations to all the participants. And we hope to have your immense participation, overwhelming participation, even for the next year. Congratulations to all of you all. Over to you, Ketki. Thank you. Vaishnavi.
Thank you so much, ma'am. Congratulations to the winners. Now moving on to day 3. I will request Dr. Tabassum Khan, ma'am, to announce the winners for UG oral presentation competition.
Okay, so the winners of the UG oral presentation competition are, so we've got a tie for the second position, and it goes to K. Gayatri and P. Saranraj team and Krishna Bhutani, Ashish Thanki, Priya Patel team. So congratulations to both the teams for this tie second prize, and the first prize goes to Rohit Pal, Jalpa Jakotra, Priya Patel from Department of Pharmaceutical Sciences, Saurashtra University, Rajkot. Congratulations on your victory to all the winners, and congratulations to all the participants as well.
Thank you so much, ma'am. Now I will request Dr. Tabassum Khan, ma'am, to announce the winners of Innovative Idea Challenge.
So the winners of the Innovative Idea Challenge, the third prize goes to Niti Madhya, Shreyans Kotecha and Parth Mishra from SVKM's Dr. BNCP. Congratulations to the entire team. The second prize goes to Advait Rao, Khushi Jain, Tian Remedios from Sunandan Devesha School of Science, NMIMS deemed to be university. Congratulations to you all. And the first prize goes to Ritika Pradeshi, Ameya Joshi, Aniket Ugale, Manthan Shirke, and Dr. Satish Polshetiwar from Dr. Vishwanath Karaj, MIT World Peace University, Pune, Maharashtra. Heartiest congratulations to all the winners in the undergraduate and postgraduate and PhD round. So congratulations.
Thank you so much, ma'am. Congratulations to all the winners. Thank you all.
Vaishnavi.
Yes, ma'am.
Vaishnavi, just a second. So, as all the BNCP team and the participants, they know that we had a UG research convention also, which was put up as a pre-SRC. It was a day before Student Research Congress, and we saw overwhelming response. The entire 117 students presented poster in the group of total 25 poster. 26 posters were presented in a group of 3 to 4 or 4 to 5 max, and the entire atmosphere of that research display, poster display area was so vibrant and wonderful. Students really enjoyed it. And all 26 posters were evaluated by senior faculty members. None of the poster had their mentor's name, and none of the poster had any of the teacher's name on that. It was purely the students who had worked on that. And that's where I just requested Dr. Sujata if we can declare first 3 top posters of pre-SRC UG research. And these 3 students will get a trophy and a certificate during our annual day program. So over to you, Dr. Sujata. Actually, I just a second before I talked to you, 2 seconds before, I just called Dr. Sujata and I asked her to quickly declare the names. So just give her a few minutes to come up with those 3 names. She has to probably open the file and look at the top 3 score and quickly present it. So Dr. Sujata, over to you.
Yeah. Sorry, we were just, all my team is also there. So we have Dr. Aarti and Dr. Atul with us. I'm sorry because it's a little bit, the room was messed up. Again, it was an overwhelming and proud moment for all 3 of us. So I'm announcing the winners. Just give us a sec. Third position, we have group number 17. The members of the group were Nidhi Rana, Shruti Rangnekar, Dharmik Rathod, Divisha Rathod, and Priyam Rawal, mentored by Dr. Radhika Raheja. At second position,
I think we are not mentioning mentor's name here. It's only
okay, fine. At second position, we have Ishan Shah, group number 19, Ishan Shah, Jash Shah, and Jinal Shah at second position. And at first position, we have group number 2, Charu Agera, Falak Agwan, Tanya Amin, Pratika Anecha, and Viral Bhanushali. So congratulations to all the winners. And we hope you continue this research for your live project. It will be a good research work.
Yes. So I request Dr. Atul to prepare a nice flyer and that we can put up on our social media for pre-SRC.
So our team is out there. Dr. Atul and Dr. Aarti
also. Yes.
So congratulations from all 3 of us to all the winners.
Thank you, ma'am. Also, I would like to congratulate the winners. So, now, thank you all for felicitating the winners and congratulations to all the winners. A note for the prize money and the certificate, the committee will contact you. Cheer to all for your diligent and earnest efforts have paid off. Also, I would like to congratulate all the participants as everyone is taking something from here, if not awards. Yet we have won the treasure of knowledge in this 3-day event. Enjoy the process of your research without succumbing to the pressure of the result. Last but not the least, I also want to thank our technical, registration, scientific, and logistic committee as the 3-day event was only possible because of the remarkable contributions made by them under the guidance of our SRC coordinator, Dr. Lokesh Bhatt sir, and the constant support of our principal ma'am, Dr. Munira Momin, and BNCP teaching and non-teaching staff. Hats off to everyone who directly or indirectly helped to make this event a grand success. Thank you, everyone. So, ma'am, with this, we would like to conclude. Okay, yeah. Ma'am, I would like to call Rashmi ma'am to give the vote of thanks.
Yes. Rashmi, unmute yourself. Rashmi madam, unmute.
Yeah, okay. Hello, good evening. Although Principal Ma'am, Vaishnavi, everybody has given a hearty vote of thanks from their end. This is an official end with an official vote of thanks. So thank you is a small word to express the gratitude we feel. I would like to extend the vote of thanks on behalf of SVKM's Dr. Bhanuben Nanavati College of Pharmacy's third Student Research Congress. I would like to extend our heartfelt gratitude to University of Mumbai and our industry partners ACG Worldwide and SciTech Center, our industrial partners for co-hosting this event. I would also like to thank our chief guest, Dr. Anuradha ma'am, and eminent speakers from ACG Worldwide who took time out of their valuable, who took time out and shared their valuable knowledge. A big thank you to our judges who happily accepted our invite to judge the event and shared their inputs. Also, many thanks to our principal, Dr. Munira ma'am, and our management who always supported us in this endeavor and motivated us to do our best. A big shout out to the organizing committee, both the teachers and students, and especially Dr. Lokesh Bhatt sir, who worked round the clock to make this event a grand success. Last but not the least, thanks to all the participants and delegates from all over India who sent their entries and made this Congress really a beneficial one. A huge thank you to one and all involved. Now, innovation is like the ocean and it never ends. We hope to see you all soon in the fourth Student Research Congress. Thank you.
Thank you, ma'am. So, thank you, everyone. With this, I would like to conclude the day 3, the last day of our third Student Research Congress. We'll be meeting you next year with the same enthusiasm. Thank you, everyone.
Should we have a group photo, students and all?
Yes, ma'am, sure.
Possible, or people have left? Let's have a group photo, whoever we have. Let's have a group photo. Yeah. Everyone, we request you to put your camera on. And yeah, you can maybe we can increase the gallery size or something like that. Yeah.
Participants, please switch on your camera.
We would like to request participants as well as also requesting Dr. Pankaj to share their views. Participants, the winners. If there are winners, we would be happy to get your inputs and also the judge, Dr. Pankaj.
Yeah, as I said, it was a wonderful experience, and it's very good that you are encouraging students to go further in the research areas. I would like to even be a spectator next time, not even judge, so that all I can see is the enthusiasm, how the students are presenting the posters and all. It was a lovely experience. Sure. They should take this further. Whoever is the winner or whoever has been shortlisted, I feel they should take this further to the next level or even closure level. It will be good for the industry as well as for them as well. Thank you so much. Thank you.
Thank you, Dr. Pankaj. May I request some of the winners, if they are still present, if they would like to share their thoughts?
Am I audible?
Yes.
Thank you so much for allowing us to participate for this very bigger event. We can say third Student Research Congress. Initially, there was, we can say, 2 rounds of evaluation. In the first day, out of 56 posters, 24 posters were shortlisted, and my poster code was PO1. So I thought I will be the starting person for the poster presentation, and actually it was a nice experience to participate in the third Student Research Congress. It was nicely organized by the team. Thanks to Munira Mumin ma'am, Principal Dr. Vanuben, Nanavati College of Pharmacy, and entire team for this wonderful event. Thank you so much. Thank you, ma'am.
Thank you. Any other winner who would like to speak? If we have here winners.
May I speak?
Yes, please.
Yeah, so this is my first time participating in this competition. One of our teammates participated last year as well. Yeah, okay, and I am so proud of my teammates. They've really done a lot of work, and I really love the experience. I'm definitely going to participate again, and I'm really looking forward to next year's competition. All the other innovative idea challenges were really good. Team 7, obviously they were the best. Like everyone knew that. Team 7 were the best.
Before participating next year, you conclude your lemonade first.
Yeah, we're definitely going to work on that. Yeah. Team 7, big shout out to you guys. You guys had the best presentation. No, seriously. Yeah, that's all I want to say. Thank you.
Yeah, so can we have Team 7 here?
Yes, we would like to thank for the opportunity of presenting our idea at this scale, and also to Dr. Pankaj for wanting to hear us out even more. Like we would love to meet with you or get in contact with you and get some more inputs on our project because we desperately need some, and plus we are trying to get funding for us, so that would be really helpful. Thank you so much for the opportunity and thank you.
Thank you. Anyone else? Student probably can put your smart board camera on, and the entire technical room would be visible, and everybody would be visible. So we can have a quick screenshot picture. Any other speaker? Maybe OPD 11, if you are here, if you would like to speak.
Hello, ma'am. First of all, I would like to thank all BNCP and SRC team for providing me such an opportunity to presenting my works, and I'm very happy to say that it is my first prize that I have won till now, and I have joined. I have joined this competition first time, and I won this prize, so I am very happy. Thanks once again.
Good. Congratulations. Yeah, over to you, Lokesh sir. The one behind the scene.
So thank you everyone to making this a grand success, and year on means it is just growing and growing. This time means it was so big we were like confused, but finally it is done and it is done good. So congratulations to all the participants and my team here. From last 3 days we are doing everything here. So it was fun. We enjoyed a lot, and the knowledge was awesome. Everything was good, and I hope next year it will be much bigger. Right, we'll come with more events and more prizes. So, and thank you very much, madam, for all the support, all the staff, right, faculties, non-teaching staff, everyone. Thank you very much. So, yeah, with this,
Big round of applause to the entire team, presenter, participants, judges, students, teachers, organizers, volunteers, everybody. Thank you.
So, with this, we would like to conclude a very successful and this grand event of 3 day of 3 day, the third Student Research Congress. And it was wonderful learning from all the presentations, and it was a wonderful event. Thank you so much, everyone. See you next year.
Students, everyone stay in your 305 room. There is a treat reaching you. So stay there in that room only. All right. Thank you. Thanks.
Thank you, ma'am.
Okay.